Next up for us is nervous tissue, one of my favorites, because it's just so interesting that everything that you are, everything that you think is based on the experiences that you've packed into this 3 pounds of pudding-like tissue that's in your cranium right now. That's pretty amazing. I also really like this topic because all of the information is relatively simple, but we can kind of put it together into a big picture. So it's one of our first times in A&P where we see the forest from the trees, where we don't just see individual facts, but you can put it together into a story that kind of makes sense. And that sense is that your neurons essentially make decisions. It divides things into pros and cons, and then pros and cons have various strengths, and then ultimately you have to make a decision based on those pros and cons. And that's what each of your billions and billions of neurons are doing. So let's pull out a little bit and see what our major topics are. So we got to do basic organization of the nervous system. The nervous system is awfully complex because you have the brain and the spinal cord and peripheral nervous tissue. So there's a lot going on. In fact, in A&P, we're going to spend a lot of time on nervous system from now on. So we're going to do nervous tissue, then we'll do the central nervous system, then we'll do the peripheral nervous system, then we'll do the autonomic nervous system. So we have a lot to do with basic organization of the nervous system. We'll talk about glia, which are the support cells. You might remember astrocytes from doing histology. There's some other ones we have to add. We'll do parts of neuron, and then here's my favorite part, is putting it all together. How do neurons function to help us make decisions? So not a whole lot of content, honestly, but it's content that you have to get to fit together. And it's a little hard to juggle it at first, but eventually you'll be juggling it and you won't know why you're confused in the first place. So let's start with basic organization of the nervous system. So using figures from OpenStax, check them out because that's a cool thing. It's a free online textbook that you can donate to support them. Anyway, the master controlling system and communication system of the body is the nervous system. So we've got the brain. We've got nerves, which are part of the peripheral nervous system. We got digestive system, nervous system, so the enteric nervous system. We got ganglia that are involved in the peripheral nervous system. And of course, the spinal cord. What the nervous system does is 3 main things. It takes in information from the outside, thinks about that information, and then puts it into a plan that we execute with motor output. That motor output can be skeletal muscles, could be glands like adrenaline, things like that too. So the nervous system has 3 overlapping functions, which the following represents a logical sequence of these 3 functions. The logical sequence that I see is you take in sensory information. Integration means you think about it. You put that information together. So, for example, maybe you put together that you felt cold, hard, about the size of a baseball, and it's dripping off of you right now, so you got hit in the side of the face with a snowball, and you need to pick up your own snowball and hit your daughter back with one. Just kidding, but not really. So simply put, the reason I like the nervous system is we make tiny little decisions. In this section, basically, learning without thought is labor lost. Thought without learning is perilous. So what I mean by that is here's a section where you really get to try and put things together and have it make sense. If you only try to memorize the facts, it's pretty boring. But the nervous system allows you to have lots and lots of aha moments, that kind of intrinsic motivation, that aha moment inside of you that says, man, learning is fun when you get to these moments where it just comes together and you're like, I feel freaking brilliant. There's a lot of that in the nervous tissue. So we're going to go through this later, but how neurons make decisions. But this is where I'm really trying to get to. So using that OpenStax figure, it was a little bit confusing, but here I'm going to break it down for you. What are all the different parts of the nervous system? So we got nervous tissue that we break down into the central nervous system and the peripheral nervous system. So central nervous system is the brain and the spinal cord. Peripheral nervous system is the spine and cranial nerve. So once you've left the brain and the spinal cord, you're in a peripheral nerve. Those nerves might be sensory. Those are nerves that are coming back into the brain. And we divide those into 2. Sensory, which is skin, skeletal, muscle. I know it's kind of redundant, sensory and sensory. But we're trying to separate things from visceral. Visceral means internal sensation. And you probably already know this, but your visceral sensation is not very good. When people have heart attacks, they feel the pain in their shoulder. When you have kidney problems, you can actually feel that in the lower back. So we don't have a good sense of what's going on inside of us. So that's why we separate these 2 systems. We have 2 different motor systems, or we can break those down into 3 different motor systems. So motor somatic is controlling skeletal muscles. And then we have something called the autonomic nervous system, which regulates muscles, cardiac muscle, and glands. So it can speed up your heart, slow down your heart. The way we break these down is into the sympathetic nervous system, which we often call the fight or flight system. Then the parasympathetic, when you want to rest and you want to digest and you're not trying to get away from something, that's the parasympathetic. So these 2 systems basically regulate energy levels, up your energy levels when you're facing a threat, lower your energy levels when you want to conserve energy. So as I said earlier, it's going to be quite a few weeks of nervous tissue right now because we have to go through nervous tissue in general. How does the neuron work? What are the support cells? Then we'll go through the central nervous system. We'll talk about all the parts of the brain and the spinal cord and what those parts do. Then we'll move into the peripheral nervous system. Honestly, most of that will be cranial nerves, so the nerves that are coming out of the brain to control things in the head and sometimes the shoulders and sometimes the heart. And then we'll move into the autonomic nervous system. The other thing that we could probably put in here is sensory. We won't spend a lot of time on sensory in AP1, but we'll spend some. So we're going to spend a lot of time on nervous tissue moving forward now. And these are just in green because I think that one big figure outlines it all at this point, but I still wanted to have this available if you wanted to see it written out. What's the somatic nervous system? What's the autonomic nervous system? All right, so that's basic organization of the nervous system, and that's, in reality, our schedule for the next several weeks. Next up is glia. A lot to do here because we divide the glia up into central nervous system glia and peripheral nervous system glia, and then we have several different cells in each of those. So what are glia? If you look at the brain, you see two main types of cells. You see neurons. Those are the excitable cells. Those are the ones that do the thinking. In this picture, it's the green cell. But neurons are pretty delicate, and they need a lot of support. They need people to clean up. They need people to basically surround them so signals get through. Those glia are the supporting cells. It's kind of interesting that people actually believe that Einstein's brain was smaller than the normal brain, but he had more glia. He had more support cells. So the 6 different types of glia, we have 4 in the central nervous system. We have what are called astrocytes, microglia, ependymal cells, and oligodendrocytes. In the peripheral nervous system, indicated here in yellow in the figure in on the left in the text, are Schwann cells and satellite cells. Again, nice figure from OpenStax. Astrocytes, microglia, ependymal cells, and oligodendrocytes. First of all, who are these glia taking care of? Got to throw this in somewhere. They're taking care of neurons. We're going to come back to neurons when we do the parts of the neurons. But we've looked at this slide in lab. The big purple one in the middle is a pyramidal cell. So this is a motor neuron cell. The little guys are astrocytes. First thing to note about neurons is they're amitotic. They don't really divide. They don't even have the spindles or the centrioles to divide. So neurons don't divide. They do communicate, and they do have 4 different parts, which we'll get to later. But let's move on to the glia cells that support the central nervous system. So astrocytes, real spiny. This guy in green is an astrocyte. Really love this figure because this red thing is a capillary. The blue guys are neurons. And one of the main things astrocytes do is they form something called the blood-brain barrier. So nothing in this blood vessel is going to make it to this neuron without going through the astrocyte first. The astrocyte chooses what comes out of the blood and makes it to the neuron. So this is the blood-brain barrier. Not everything in this blood is going to make it to this neuron. The astrocyte is the gatekeeper. It's one of the main things astrocytes do. So that's where down here it says controls the chemical environment. So it basically controls what makes it from the blood into the neuron. They're also support. They're like a scaffolding for the neurons to sit on. So you can kind of see these green fibers running through here. That's the scaffolding that the neurons kind of hang off of. Like I said, they're the bridge between neurons, and they also will guide migration of young neurons. So when you're growing, you need to have a neuron leave your brain and go down through your spinal cord and find the meeting place for another neuron that's going to go down to your big toe so you can wiggle your big toe. You need guideposts along the way, and that's what astrocytes do. Ependymal cells, pretty simple. You have these ventricles in your brain. We'll talk about those. They're the blue areas down here. Sorry, it's kind of low resolution. But your brain is actually floating because there's these ventricles that hold water, or CSF technically. Lining that CSF are ependymal cells. So they line the ventricles of the brain and the spinal cord. Microglia are your brain's immune system. So these are some real ones. The microglia is shown here, and this is kind of a cool figure because the neurons are in blue, the astrocytes are in red, and the microglia are in green. So just imagine you want your own little cell to roam around the brain and make sure no bacteria is in there. That's what the microglia does. Oligodendrocytes, they provide insulation for neurons. We'll talk about this in a second, called myelination. So your neurons carry electricity. And just like the wires in your wall have insulation so they don't short circuit, your neurons actually have to be insulated against short circuiting. And that's what the oligodendrocyte does. You have a different cell in the peripheral nervous system, and that actually has some connotations when it comes to healing. So oligodendrocytes don't heal very well. So if they're disturbed, if they're no longer wrapping a neuron, they can't find that neuron again, which means that neuron's not going to function again. But Schwann cells, the other type of cell that's in the peripheral nervous system that does myelination, heals much better. So if you cut a peripheral nerve, it might heal back. It might grow back. if you cut a CNS neuron, because of the damage to the oligodendrocytes, often that neuron is cut forever. It's not going to heal. It's kind of a cool structure here. So the oligodendrocytes are these guys. So this might be the soma. They reach out these little hands that form myelination over the axon of a neuron. So that's the communication part of the neuron. Maybe come back to this after we've talked about parts of the neuron, and this will make more sense. But neurons actually have electrical conduction systems that jump down the axon. In certain places, you have to have gaps. We'll talk about those as nodes of Ranvier. But other than that, you need a place where things can't leak in and out, and the oligodendrocyte covers up the neuron, covers up the axon, so things can't leak in and out. This is a really interesting figure here to me because the orange are the oligodendrocytes. And you notice in certain places there's a gap. That gap would mean there's something wrong with this oligodendrocyte. Do you know the condition where you get gaps in your myelination? It's multiple sclerosis. Multiple sclerosis is when the immune system attacks oligodendrocytes, so then you have gaps along the axon. And it can lead to short-circuiting. And it can also really stress the cell because ions are leaking out. And remember, the sodium-potassium ATPase is charged with keeping ions in the right place. So it's really hard on the sodium-potassium ATPase to keep ions in the right place if the neuron is leaking because the oligodendrocyte is missing. And that's where I put in a couple of slides down here. MS, it's autoimmune disease, mainly affects young adults. One of the interesting things about it is it can come and go. Often it goes away when a woman is pregnant. So we really don't understand the process, but basically what happens is the nerve impulse short circuits. There's a lot of different treatments. The main one is to shut off the immune system. So we've talked about interferon. Interferon will calm the immune system cells so that they don't attack those oligodendrocytes as well. Anyway, that's MS, and that's related to oligodendrocytes. Those are the glia of the CNS. We have PNS glia, or peripheral nervous system glia. The two ones here are Schwann cells and satellite cells. I don't think we really need to skip off this slide, so let's just look at them here. Schwann cells, kind of like those oligodendrocytes, provide myelination. And again, they create gaps because we need sodium ions to come out here and potassium ions to go back in here. What the Schwann cell will do is it's not like an oligodendrocyte where it reaches an arm out and myelinates. One Schwann cell, shown down here, will wrap around and around and around the neuron. That's kind of interesting because if you were to cut this axon, the Schwann cells usually stay right in place. So when the neuron tries to regrow, it will regrow through that tunnel that was left by the Schwann cells. If you think about oligodendrocytes as extending these arms, when they extend their arms, if the neuron disappears, then the arm kind of shrivels up and it doesn't stay in that place and leave a tunnel. So this is a main reason. If you cut a peripheral nerve, it will often heal back. But if you cut a central nervous system, so talking about spinal cord injuries, the nerve can't grow back. Okay, satellite cells are these guys up here that surround the soma, or the cell body of the neuron, and provide support there. I guess I did make another slide, but it's kind of the same slide, so kind of ridiculous. So that's glia. Next up is parts of the neuron. Pretty simple here. 4 different parts of the neuron. Soma, which is the cell body. Dendrite, which is where information comes into the neuron. Axon, which is where the neuron relays information down to a target that's further away. And synapse, this is where one cell will talk to another cell, or where one neuron will talk to another neuron. Let's break those down a little bit. So synapse is where 2 neurons will join each other. So it's a junction that allows information transfer. And when we get over to our umbrella metaphor, we'll talk about this in terms of usually a neuron, not always, but usually a neuron will release neurotransmitters. Sometimes it's just an electrical current, but most of the times you're releasing a neurotransmitter. What has to happen is you have to get the voltage at the end of the cell to plus 30 millivolts. Voltage channel, calcium-gated voltage channels will open. Calcium will come in and cause the neurotransmitter to be released. Again, we're going to talk about that later, but now we're talking about the cells, just kind of previewing what this part of the neuron does. The axon relays information. A decision usually occurs here in the axon hillock, but it has to relay that information to a far-off target. So again, think about if you want to wiggle your big toe, the decision is made up in your brain, but the signal has to be sent to the muscles that are going to wiggle the big toe. So you need an axon to do that. Technically, it takes 2 axons. One goes down through the spinal cord, and then it will synapse. We just talked about synapses. It'll synapse, and another neuron will go down to the muscle that wiggles the big toe. So a lot of times the decision is made a long way away from the target, and so the axon is what carries the signal from where the decision was made to where the target needs to be moved or needs to be activated. Sometimes you call the end the axon terminal or the synapse. Myelin sheath, mentioned already that if you have an electrical wire going through your house, you need to insulate it to make sure that you don't have short circuits. So showing this kind of actual telephone wire up here. Real neurons look very similar because real neurons, the orange guys are the neurons and the white is the myelin. So the myelin sheath in the central nervous system is provided by the oligodendrocytes and it's provided by the Schwann cell in the peripheral nervous system. So what it does is it protects the axon. It insulates the fibers electrically, and it increases the speed of nerve impulse transmission. It's kind of interesting, we'll talk about this later, but not all neurons have myelination. What's interesting is neurons that carry pain are often unmyelinated, but neurons that carry touch often are myelinated. So if you go over to the wall right now, I'll probably make this joke later, if you go over to the wall right now and kick it really, really hard, you'll feel that you touch the wall about a half second before it's painful. Again, that's because touch is on a myelinated fiber and pain is not on a myelinated fiber. Another interesting one there is when you decide to go to the bathroom, like we're talking about urination, when you decide to urinate, it takes about a half second before you actually initiate flow, and that's because the axons that are relaying the information are not well myelinated. Just trying to point out that some signals happen really fast because of myelin, and some signals happen kind of slow because they lack myelin. We'll learn about why this is important, but those little gaps in the myelin are called nodes of Ranvier. And once we talk about action potentials, it's going to be evident why those are there. So here again, myelinated versus unmyelinated. The guys that have purple are myelinated. The ones that don't have a lot of purple are unmyelinated. The other reason to point out myelination is it gives the basis of white matter versus gray matter. So you've heard use your gray matter. Your gray matter is the soma of neurons. It's where the decision occurs again. White matter is where the signal is relayed. White matter is white because that myelination or that fat is white. It's interesting that in the brain, the gray matter is on the outside. It's a very thin little, it's like if you had an orange, the cortex of the orange would be the thinking parts. The cortex of your brain is only 5 to 6 millimeters thick, and that's on the outside. Everything else is white matter, and this is just where neurons are talking to other neurons. It's interesting that the spinal cord reverses things. The white matter is on the outside, and the gray matter is on the inside in the spinal cord. So that's the axon. Dendrites are where a neuron takes in information. So when you're going to make a decision, you got to weigh different options. So if this neuron is going to make a decision, it has to receive inputs on why I should make this option, this pro, this con, or whatnot. The dendrites bring in information to neurons. Last is the soma, or the cell body. So every cell has to have a place that has rough ER and has mitochondria and things like that. It's interesting that neurons, they have such a developed rough ER that you can see them pretty well in a microscope, and they've been called Nissl bodies. It gives us an indication that neurons use a lot of protein, whether that's how we store memories or what it's for, but neurons use a lot of protein. So the soma is basically the same in a neuron as every other cell in a lot of ways, in that it has a nucleus and a nucleolus, and it has rough ER and smooth ER. It's a little bit different than other cells in that it has this focal point, this axon that grows out of it. Mentioned this before, it has no centrioles, so it can't go through mitosis once it reaches a certain age. It has something called an axon hillock that we'll talk about soon, and that's essentially where decisions are made in a neuron. So that's the soma. Now, if you start looking at these different parts, they're kind of arranged differently on different types of neurons. So we have what are called multipolar neurons. These are the most predominant neurons. They have lots and lots of dendrites, and usually a single axon that extends out to a target. That's called a multipolar. That's different than a unipolar. A unipolar might have input at one end and just an output at another end. So not a whole lot of dendrites, just kind of one signal. And in fact, most of the time they're like this, where the soma is out of the way. The critical difference here is this guy has an axon hillock, so multipolars have an axon hillock to make decisions. Unipolar gets the soma out of the way. It doesn't make any decisions. Once there's a signal generated at one end, it goes to the other end. This is primarily used in sensation, so there's no need to make a decision about whether you convey to your brain that you just kicked a wall. You definitely want that signal to go to the brain. And so you have the nucleus off to the side, you have the soma off to the side, so it's called unipolar. Once in a while, that sensory information is really complex, and so you do some pre-processing. In this case, it's called a bipolar neuron. So bipolar might receive a lot of information, have a lot of dendrites, and a lot of axons. So it's going to process information. So in this case, it might have a lot of dendrites and have a lot of synapses to relay complex information. So let's hit that again one more time. Multipolar, there's a lot of processing. Take in a bunch of information, make a decision, output that information. That's multipolar. That's most neurons. Unipolar, just even get the soma out of the way. Once information comes in one side, it goes to the other. That's sensory. Bipolar is when you have complex sensory. So your eyes do a lot of processing. They help you figure out lines and shades and things like that before it ever gets to the brain. And it's because you have bipolar cells that take in a lot of information through a lot of dendrites and can form a lot of synapses too. So functionally then, we just talked anatomically, what's the different structure. Functionally, we have 3 different types, which goes back to what we initially said. The brain does 3 things. It does motor, it does processing or association, so those are interneurons. And then it does sensory, it has sensory. So in general, motor are always multipolar. Interneurons are always multipolar. Sensory, most sensory, like for touch and things like that, are unipolar. Then you have these specialized bipolar cells that carry information on smell and on vision. So let's just try and make that more clear. Multipolar, motor neurons, interneurons. Unipolar, sensory but simple. Bipolar, sensory but complex sensation. So that's the link between what anatomical type of neuron you have and what that neuron is supposed to do. So those are types of neurons. Now is when it gets interesting because we have to start thinking about how these cells actually work. I really like something called the umbrella metaphor. A slight backstory is when I graduated from school, I stayed home with my kid for 3 years, which was just awesome. Before that time, if I was going to have to take an umbrella somewhere, I never took an umbrella. I never, ever took an umbrella. But one day I took my daughter out. She was about 2. And I was like, well, we'll just be tough. Well, it did not go so well. My daughter did not like at all being rained on. So I learned at that moment that from now on, if I'm going to go out in the rain and Ella is with me, I need to take an umbrella. So my neurons basically had to learn and change their decision process. So before Ella, the decision process was always no umbrella, no reason to take an umbrella. Once I had Ella, it became once Ella is in the equation, I will always take an umbrella. So just going to go through how my neurons make that decision and how they changed that decision waiting when Ella was brought into the picture. So the questions we need to ask: what makes the electricity flow in the axon? What happens at the end? How do dendrites govern? These are essentially the questions that I've come up with. What are the initial conditions who sets up neurons to be excitable? Why do ions want to move? How do ions move? What happens when ions move? How does fast signaling occur? How do you get different strengths of input? So maybe there's other reasons to take an umbrella, but they're kind of weak reasons. How do I all of a sudden have this Ella that's a really strong input to tell me to take an umbrella? There's lots of pros and cons in taking an umbrella. Well, you got to carry it around. Well, I don't know where it is, and things like that. Those are all cons. Pros are maybe I'm carrying my computer. Maybe I have to park a long way away from the building. Maybe it's really going to be a strong storm. Things like that. So there's different pros and cons. Once you make a decision, how do you tell your brain to say, hey, where's my umbrella at? Neurotransmitters and methods of processing can shape that decision. So I said this at the beginning. The reason I want to do this is because it's not because I'm mean, but it is because drugs affect ion channels. Understanding action potentials will help you understand how the heart works. And there we definitely give drugs to affect the heart. There are conditions that arise from problems with ion channels that we'll need to basically understand, or you should understand when you head into healthcare. Okay, so now that I read all those off, let's go through them one time really quick, just to kind of introduce each step, and then we'll go through each step with a little bit more detail. Finally, there is one long video out here. It's called The Umbrella Metaphor. You can find it on YouTube. They will line up everything in 3 passes of the story and basically talk about how neurons make decisions. But first, what are the initial conditions? You have to set up a condition where you can use electrical signaling, and that's set up by the sodium-potassium ATPase. So you end up with a negative charge inside the cell. We're going to change that negative charge back and forth so we can signal fast. Why do ions move? Just pointing out that if you pump a bunch of sodium outside the cell, the sodium's going to want to move in. And since it's positively charged, when it moves in, it's going to change the charge on the cell. technically there's that concentration gradient, but there's also an electrical gradient too, because since the inside of the cell is negative, sodium wants to come in because of its concentration gradient, but also because the inside of the cell is negative, which is important to know because potassium wants out of the cell because it's concentrated on the inside of the cell, but it kind of wants to stay in the cell because the inside of the cell is negative and it's attracting the positive charge of potassium. How do ions move? So we established that ions want to move. Well, can they move? Can they move in and out of the cell? They can move with the aid of ion channels. And there's 4 different types of ion channels. 2 are important in this drawing. So those are voltage gated and chemical gated. And you'll see when we draw the cell out, those ions will come up. Those ion channels will come up. There's 2 more that will come up later in AP1. Leak gated channels are important in heart function, and mechano-gated are important in sound. So that's how ions move. What happens when ions move? Don't need to make this any more complex, but just pointing out that this cell is sitting at minus 70 millivolts. If all of a sudden you let sodium come in, it's bringing a positive charge with it, so the inside of the cell is going to get more positive. It's a little bit harder to see, but if you open up a channel and let potassium out, you've got a positive ion leaving the cell. That means the outside of the cell is becoming more positive, which means the inside of the cell is becoming more negative. So what we have here then is by letting ions move, we can change that voltage on the cell. And that's how we signal things quickly. The way we draw it up, not to get into too much detail yet, is we call it an action potential. We'll come back to this, but essentially when you change that voltage from its normal minus 70 up to plus 30 and then back down, this is kind of like alternating current. It's kind of a signal. And so this is the way when we want to wiggle our big toe, we can get that done in 0.3 of a second, is we use these things called action potentials. And this is the way we do fast signaling. When you're making a decision, though, you have various strengths of input. So maybe having Ella with me is a very strong reason to take an umbrella. Maybe carrying a computer or knowing that the parking is going to be bad is as strong, but not as strong as Ella. So I have various strengths of inputs. I also have positives and negatives. So I have reasons to take an umbrella that may be strong and may be weak, and I have reasons not to take an umbrella, and those might be strong and weak. So how does my brain put those all together? So how do you get pros and cons? So I might take an umbrella because Ella's with me. I may not want to take an umbrella because all I have is a Hello Kitty umbrella, and I don't want to be caught dead with a Hello Kitty umbrella. So I've got pros and cons. How does a neuron weigh pros and cons? And then when those pros and cons have different strengths. Let's say the neuron has decided to carry an umbrella. Well, how is it going to relay that information to the part of my brain that says, hey, this is where your umbrella was last time? So that's synaptic transmission. So once a neuron has made the decision to tell another cell to do something, how does it tell it? Neurotransmitters are the normal signal that neurons use to talk to other neurons. So this neuron that has to tell this other neuron, try and remember where your umbrella is, it has to tell that neuron through neurotransmitters. There's some neurons that talk with just electricity, but most use neurotransmitters. The other reason to know this is because we can give drugs that affect this process. At one time in this country, 1 in 5 people, it might have even been 1 in 3, but I think it's 1 in 5 people were on selective serotonin reuptake inhibitors. And that term will make a lot of sense once we talk about neurotransmitters. But those are antidepressants that make it harder for cells to get rid of serotonin in the synapse. So we give drugs to affect neurotransmitters. Then we have methods of processing. We won't dwell on this a whole lot, but neurons can arrange themselves in different ways in order to make different types of decisions. So we'll talk about that. All right, getting into a little bit more detail, what are the initial conditions and who sets them up? So if you want, you don't necessarily need to listen to this. You can skip to the one video that's out here, and it's on YouTube. And it'll go through all these
Our next major topic is going to be nervous tissue. And the way I like to teach,
it'll go through all of these questions on its own. But maybe you want to hear them a couple of times. And so we can do this individually too. So the initial conditions are set up by this guy, the sodium potassium ATPase. And it sets up 2 conditions. It sets up a chemical gradient. So it's going to pump 3 sodium out, 2 potassium in, and burn an ATP. That means it's going to set up a chemical gradient where sodium is going to be on the outside and potassium is going to be on the inside. You're also setting up an electrical gradient. Now, the simple way to say this is just, well, you're pumping 3 positive charges out and only 2 positive charges in, so you're going to get a buildup of positive charge on the outside. Now, the first guy that talked about this, he could have said the outside of the cell is positive relative to the inside, but he said, I'm going to talk about the inside of the cell as negative. So the inside of the cell is negative simply because you're pumping more sodium ions or more positive charges out and less are coming in. The technical way that this gradient actually develops is sodium and potassium can both leak through the cell membrane. Potassium leaks faster out than sodium leaks in. So you are getting a little bit of charge, maybe 15% of your negative 70 is by pumping sodiums out and only taking 2 positives back in. But most of the charge is developed because then the potassium is allowed to leak out, making the inside of the cell even more negative. Kind of a technical issue, but you really just need to know that the inside of the cell goes to minus 70 millivolts, and we have 2 gradients built up here. We have a chemical gradient, that's where you have more sodium on the outside and more potassium on the inside, but an electrical gradient where the inside of the cell is more negative. So those are the initial conditions. What I would recommend is take 15 minutes and just get that. Maybe that clicked right away and it makes perfect sense. Maybe it didn't click and you need to take a break after 15 minutes and come back to it. Because then you'll want to add on. So we're going to build information, and if you don't get that first step, then step 2 is not going to make sense. So make step 1 make sense and then come to step 2, which is, again, is kind of a simple one, which is why do ions move? So we established that these ions are pumped out or pumped in. They're going to want to move. Now the first one is obvious. That chemical gradient is set up so that sodium is packed up on the outside of the cell. Sodium likes to get away from other sodiums by diffusion, so it wants to go into the cell. Also, potassium doesn't like being packed inside the cell, so it wants out. That's the chemical gradient. But don't forget this minus 70. This minus 70 also attracts the sodium in. Technically, it makes the potassium want to stay. So the electrical gradient makes sodium want to move in because it's following both its concentration gradient and its electrical gradient. Technically, potassium wants to leave by its concentration gradient because it's concentrated on the inside, but it doesn't want to leave because it's attracted to the negative 70 that's inside the cell. Overall, the chemical wins. That's why if potassium can leave, it will. It'll just leave slower than sodium would be allowed to come in. And in fact, there is a channel in muscle that it just opens up a hole and lets sodium come in and potassium leave through that same channel. Sodium comes in faster because it's following both its chemical gradient and its electrical gradient than potassium leaves. And so you end up making the cell turn positive in order to stimulate muscle contraction. And that's how muscles contract, or that's how something called the nicotinic acetylcholine receptor works when a motor neuron tells a muscle to contract. Opens up a hole, lets sodium and potassium flow. Sodium comes in faster than potassium leaves, and you depolarize or activate the muscle. Don't really need to get into this in too much detail. Just know the terms voltage is the measure of potential energy. So we talk about the inside of the cell as having a negative 70 millivolts. Current and resistance are part of Ohm's law. We'll talk about these later on, ironically, when it's not about electricity but flow of air. But I don't know that we need to particularly get into detail. I mean, you might see Ohm's law. Ohm's law is a good law to know. Again, it's going to come up. And it's basically the current, how fast something moves, is dependent on how much it wants to move, divided by what's in the way. So Ohm's law, how much something will flow, is dependent on how much it wants to flow. In this case, voltage drives current flow, divided by resistance. And we'll talk about Ohm's law when we get to blood flow and airflow in the lungs. So again, the first step here is just to set everything up with the sodium-potassium ATPase. So that's why ions move. How do they move? So there's 4 main types of ion channels. There's voltage-gated, where those depend on the voltage of the cell to open and close. Ligand-gated, where there's some chemical that opens the channel. Mechano-gated, there's a bit of force that opens the channel. Or leak channel, they're just open all the time. Voltage-gated, the way they work is they have a positive charge attracted to the gate. So here's the gate closing the channel, and it's attracted and it's attached to some positive charges. If the inside of the cell is negative, then these positive charges are attracted to the inside of the cell, and the gate stays closed. Let's say the inside of the cell goes up to plus 30 millivolts. Well, now those positive charges are not attracted to a negative inside of the cell. The inside of the cell is not negative anymore, so the gate slides open and the channel is open. With ligand-gated, it really comes down to if there's a chemical bound, the channel is open. If a chemical is not bound, and it's a specific chemical, like a neurotransmitter, if it's not bound, then the channel is closed. Leak channels, really small. We'll see these in the heart. They're just open all the time. Mechano-gated, we'll see these in hearing. If there's a force pressed on the gate, in this case, it'll be sound wave, then the gate will open and sodium will flow. So that's how ions move. There's different types of channels. Just kind of already went through those. So once these channels are open, ions move quickly. Ion channels only really open one ion through. Mention the one in the muscle that lets sodium and potassium flow together. That usually doesn't happen. Usually there's only a channel that lets only sodium through or only potassium through. So these are just some pictures of the different types of channels. So passive or leaked, really small ions just flow through. Chemical gated, something binds, like in this case acetylcholine is the neurotransmitter that causes muscles to contract. When it's bound, the channel opens and ions flow through. Mechano-gated, usually there's some kind of gate that's pushed on by force, sound wave, and that will open the channel. And the last one is voltage gated. It's not shown as nice here as I would like to show. Is literally there is a gate that's got positive charges, and that positive charge slides in and out relative to the voltage inside the cell. When the inside of the cell becomes less negative, the gate slides out and the channel is open. So that's the operation of a voltage gated channel. I like to throw this one in just to show a little bit of physics, but you don't really need to know the Nernst equation. But essentially what it's talking about is a certain voltage will develop in the cell based on the concentration of ions. So don't need to get into the detail, but the concentration of sodium on the outside versus the concentration on the inside will very specifically set up one voltage of negative 55 millivolts, negative 30. So it really all does come down to the concentration of ions. Ultimately, you end up with a resting membrane potential. This gets kind of confusing, but the inside of the cell is called the VM. So the resting membrane potential is called the VM, or sometimes the VR. The VM is usually around minus 70 MV for millivolts. All right, so that's how do ions move. Kind of another simple one, what happens when ions move. And this is just to point out that when sodium enters, it's got a positive charge. So it's going to make the inside of the cell go more positive. When potassium leaves, a positive charge is leaving the cell, so the inside of the cell is going to become more negative. That's kind of one that like clicks right away for some people, but some people it doesn't click, and you just need to keep thinking about that. If sodium comes in and it's positive, then it's going to make the cell more positive. Another tricky one is sometimes you can have negative ions. We'll get to this when we get to those cons, when you don't want to take an umbrella. So what can happen then is chloride can enter the cell. And chloride is negative, so when it comes in, it makes the cell more negative. So we have this potential set up, and it's going to be affected by ions moving in and out. A couple of terms is when a cell becomes more positive, it's called depolarization. So usually a cell sits at minus 70 millivolts. That's called polarized, as in like the Earth has its poles because the north and the south pole are different. A cell that's negative on the inside has a polarity. It's more negative on the inside. So if we remove that polarity, if we depolarize, it's because we've made the charges more even, or we've made the charge closer to zero. So depolarization is when voltages go back up to zero. Repolarization is when we redo that polarization. We make the voltage go back down to negative voltages. Then we'll see that sometimes a cell can become hyperpolarized, more negative than it normally would be. And this is just to show how we measure this. We can literally put little glass electrodes into neurons and see that the outside is more positive than the inside. I used to do that. Not in neurons, but in cells. So how does fast signaling occur? At this point, hopefully you're realizing, well, yeah, I can kind of change the voltage by letting sodium in or letting potassium out. The way we draw that is on a scale with time on the x-axis and voltage on the y-axis. And I don't know if you can see these, but I've written down here, or it's on the bigger drawing, that right here, all the channels are closed, so we're at resting voltage. Well, what would happen if we opened up a voltage-dependent sodium channel and sodium came into our neuron? Well, then our voltage is going to go up, or we're going to depolarize. At the top, sodium channels have something called an inactivation plug. It plugs the hole, and so this channel is closed. At around plus 30 millivolts, potassium channels open and potassium leaves the cell. If potassium's leaving the cell, then the cell is becoming more negative, and so that's why our voltage comes back down. Usually those potassium channels stay open a little bit too long, and so they let the voltage drop back below rest. This is called hyperpolarization. At that point, then the voltage-dependent potassium channel will close. All the channels are closed again, and so we're back at rest. So a couple of things to get on here is the shape of the voltage changes. That's the action potential. To know that this is resting potential, This is depolarization, this is repolarization, hyperpolarization, and back to rest. So when we talk about how does fast signaling occur, we're just talking about how does the cell generate an action potential. So what an action potential is, not to just read these off, is it's a reversal of memory potential. You've gone from a negative 70 up to a positive. And this is really how you get a signal to move quickly. So mentioned this already, we've got 4 phases: resting, depolarization, repolarization, and hyperpolarization. Each of those phases is determined by different channels. So again, rest, everybody's closed. Depolarization, the sodium channel is open. Once we get to the top of the action potential, sodium channel is closed, the potassium channel is open. Potassium leaves the cell. When potassium leaves the cell, we get more negative and our voltage comes down. Those potassium channels stay open a little bit too long, and so we get a hyperpolarization. Once the potassium channel closes, what actually regenerates our resting potential is the sodium-potassium ATPase, brings our voltage back up to minus 70, and then we're done. We're going to wait for a signal again. I think I've talked through each of those on one figure. So depolarization, again, it's all about the sodium. Takes you from minus 55 millivolts. We'll talk about how did you get to minus 55 millivolts in the first place. But you'll notice here we're at minus 70. When we get to minus 55, that's when the sodium channel opens. Repolarization is driven by potassium channels. The sodium channel has been inactivated. And then hyperpolarization is when those potassium channels have stayed open too long. Then the sodium-potassium ATPase brings us back up. So just another view, I think maybe a close-up view that A, everybody should be closed. B, sodium channels open, potassium's closed. Repolarization, sodium is inactivated, potassium's open. Once we're through the undershoot, we go back to rest. You might ask, with these action potentials, well, how then do I determine how strong a stimulus is? Are there big action potentials? Are there little action potentials? And the thing is the coding for stimulus is in how fast your action potentials are. You make one action potential. When you open up sodium channels, they all open. So you can't get a super duper action potential. So if you want to code for, I feel somebody touching me. Oh, I feel it. That hurts. Brother's giving me a snake bite. That really, really hurts. You code for that with how fast your action potentials are generated. 2 key things: something called absolute refractory period and relative refractory period. Now, as I said, when your sodium channels are open, they're all open. That means there's no way that you can get an action potential that's bigger. All of them are open already. You can't get a bigger one. So this means once a neuron starts to be stimulated, it's going to be stimulated all the way, and you can't get a bigger action potential. Now, once the sodium channels close, technically you can get an action potential that will rise up again. This is how you get stimulus intensity. So there is something called a relative refractory period. Relative refractory period means you could get another action potential even though you're repolarizing, but it would take a super duper stimulus. And the reason this is because you could get a certain number of sodium channels open, and then if the stimulus is really strong, you could get more and more to open. But it doesn't happen often. So the relative refractory period, sodium gates are closed, they won't open. Potassium gates are open. What really happens is some of the sodium gates might still be open, but some of the potassium gates are open too. So every time a sodium comes in to re-stimulate, a potassium can leave, which means it's going to take a whole lot of sodium channels open to get the action potential started again. Conduction velocities, don't think we'll really talk about this too much, other than that the bigger the axon, the more myelin, the faster the action potential can flow. Saltatory conduction is because you've got these oligodendrocytes. You've got these spots where sodium and potassium can enter, where sodium and potassium can enter and leave the cell, let's put it that way. This allows the action potential to jump from one node of Ranvier to another node of Ranvier. The other way to think about it is there's no sodium and potassium leaking where there's an oligodendrocyte. So it allows the current to jump. But it also means you don't have to worry about sodium and potassium leaking out and lessening your action potential. So that's how does fast occur. How do you get various strengths of input? So when I decide I want to take an umbrella, maybe I have Ella, and that's a really strong positive. Maybe the only umbrella I have is a Hello Kitty umbrella. Maybe my mother-in-law says I should take an umbrella, and that's a weak positive. How do I do that? How do I have strong and negative inputs. It really comes down to Ohm's law. So all of these different inputs are going to weigh into the axon hillock and see if the axon hillock will get to minus 55 millivolts and stimulate an action potential. What that really means is sodium is going to come in through the pros, chloride might come in through the cons, and potassium might leave with the cons to make things negative. So it all depends on how much sodium, potassium, and chloride will affect the voltage down here. Those ions are going to be able to flow down here with different reasons. So the closer the synapse is to the axon hillock, the sodium that comes in is going to be right near the trigger zone. And so that's going to be a strong pro. The wider the dendrites. So here we're on the soma. So this negative has strong input because it's pretty close to the axon hillock and it's on a nice big dendrite. As opposed to mother-in-law out here where sodium is going to want to come into this axon, but it's going to have to filter through this narrow little tunnel. So by the time that sodium gets down here, it's in a single file. It's been spread out. It's not as likely to cause a positive input on the axon hillock. The other thing that neurons can do is they can change how many channels they activate by releasing more neurotransmitter. So maybe Ella really causes a lot of sodium to come in because it activates a lot of sodium channels. So the first thing to note is at one end of the neuron, out here, you're getting what are called graded potentials. Graded potentials mean the ions come in simply spread out. And that's different than action potentials. An action potential is where you regularly have sodium coming in and kind of rebooting the signal. So sodium comes in, and if it makes it down here, it activates more sodium channels. And when it activates more sodium channels, more sodium comes in. And then it goes down to the next node, and it activates voltage-dependent sodium channels. And those voltage-dependent sodium channels let more sodium come in. And so we get this reamplification as we jump down. So the difference between a graded potential and an action potential is the presence of voltage-gated channels. Voltage-gated channels will continue to boost the signal. Whereas graded, it's just kind of a mush, an addition of a bunch of negatives and positives. So graded potentials are simply the addition of the positive and negative ions that enter the dendrites. So here's a figure from OpenStack. So a small stimulus might give you a little bit of a potential, and if you add up multiple ones together, maybe you'll get a potential that will raise you above threshold. So maybe you have a certain amount of sodium that comes in for one reason through one pro and another pro, and those add up to get you enough to activate the axon hillock. How do you get pros and cons? Honestly, you get pros and cons. If we look at all these little axons out here, the way you get pros and cons is if you want to have a pro, you want to get the voltage closer to threshold at the axon hillock. So essentially what you do is you let sodium in. If you want to have a con, what you want to do is you want to keep the voltage of the axon hillock away from minus 55 millivolts. So you want to make the voltage of the soma more negative. You do that two ways. Either you let chloride in. So you let chloride in, and chloride's a negatively charged ion. I don't know why there's a positive there. It should be negative. Or you let potassium out. If you let potassium out, that also makes the soma more negative. And so there's two ways to make the voltage more negative. So pros and cons come down to, well, I need to make the axon hillock minus 55 in order to activate the axon hillock. Pros will get me closer to that minus 55 by letting sodium in. Cons will keep me from getting to minus 55 by either letting chloride in or letting potassium out. Synaptic transmission. Once you've made the decision to actually have the axon hillock fire an action potential, it needs to send that signal to other neurons through something called synaptic transmission. If we make it really simple, the three steps are we got to get the axon to plus 30 millivolts. There are voltage-dependent channels in the axon that will allow calcium in. Calcium binds to neurotransmitter vesicles, and that calcium causes the vesicles to open up and release the neurotransmitter. So it's not much more complex than that. Get to plus 30, that activates voltage-dependent calcium channels. Calcium comes in and binds to neurotransmitter vesicles that are waiting to be released. That calcium signals causes the vesicles to be released and that neurotransmitter to be released into the synapse. We have two different terms. So we have presynaptic neurons and postsynaptic neurons. So in this case, this would be the presynaptic neuron. This would be the postsynaptic neuron. And this is the synapse. So might hear those terms presynaptic and postsynaptic neuron. Sometimes you can have different types of synapses. So an axon can bind to a dendrite, or it might bind to the soma So it might bind to the soma directly. As we talked about before, if it binds to the soma directly, it usually has more power and influence because it's closer to the axon hillock. Whereas if it's on the dendrite, it's weaker because its signal has to go all the way through the dendrite to reach the soma, which is the signaling area. You can have electrical synapses. We just have gap junctions and let the sodium flow straight through or the calcium flow straight through from one neuron to the next. Now we have two different regions when we talk about chemical synapses. We have the axon terminal, and then we have the receptor region. If we look close, so this is the presynaptic neuron, postsynaptic neuron, and this is the receptor region. With pros and cons, technically pro is called an EPSP or an excitatory postsynaptic potential, and a con is an IPSP or an inhibitory postsynaptic potential. So again, an EPSP will raise voltage towards threshold, an IPSP will decrease voltage to keep us from getting to threshold. EPSPs are sodium channels, so sodium comes in, sodium's positive and raises the voltage. IPSPs, either they let chloride into the cell, and that brings negative inside the cell, or it lets positive potassium out, and that also makes the inside of the cell more negative. So IPSPs make the inside of the cell more negative, further away from the threshold of minus 55 by either letting chloride in or letting potassium out. Eventually what happens, this is a nice figure from OpenStax, is you have these different pros and cons. They're all kind of adding together. So sodium's coming in here, sodium's coming in here, sodium's coming in here, but chloride's coming in here and potassium's leaving here. That whole math of negative ions coming in, potassium leaving, sodium coming in, has to add up to a charge of minus 55. If it does at the axon hillock, then you're going to release an action potential. Then you get down to the synaptic cleft, where you'll do synaptic transmission. So again, the axon terminal gets to plus 30, voltage-dependent calcium channels open, calcium comes in, calcium binds to these little vesicles that are ready to release neurotransmitter, and when the calcium binds, they open up and release their neurotransmitter. Kind of a cool figure. So this is pre, and these are those neurotransmitter vesicles, and this is the synapse. So these guys are ready to release their neurotransmitter if they're just exposed to calcium. Okay, so that's it for synaptic transmission. Now we got to move on to neurotransmitters. The first thing we'll talk about is neurotransmitter clearance. So when you release neurotransmitter, you should release enough neurotransmitter to bind each postsynaptic receptor once and only once. That means whatever neurotransmitter is left over after all the receptors are bound, it needs to be cleared. One of the main ways it's cleared is reuptake. So the neuron will take it, the presynaptic neuron will take it back up. And that's kind of what happens normally because it takes a lot of energy to make neurotransmitters. Why waste them? You can use drugs to actually interact with this. So SSRIs are selective serotonin reuptake inhibitors, and those are antidepressants. In certain clefts, especially in muscle, you have an enzyme that breaks down acetylcholine and gets rid of it. Often, too, some of the neurotransmitter just diffuses out and it's picked up by blood vessels and astrocytes, and it's cleared from the cleft that way. So again, a little bit prettier of a picture. The three ways neurotransmitter is cleared is reabsorption by the presynaptic terminal, so there's pumps that will pump it back up. There's enzymes that might degrade the neurotransmitter, and then some of it might diffuse away, be picked up by the astrocyte and delivered to the blood vessels. There are diseases that affect the degradation of enzymes, so if this was a motor neuron and this was a muscle, this would be a nicotinic acetylcholine receptor. The nicotinic acetylcholine receptor is sometimes attacked by the immune system, leading to a condition called myasthenia gravis. One of the treatments for myasthenia gravis is to disable or to slow down the enzyme. So the immune system has taken away some of these receptors, but if the drug is given to take away some of these enzymes, then when the neurotransmitter is released, it will stick around longer. So even though there's fewer receptors, it will keep stimulating those receptors in order to bring back some of the muscle strength. That's just another termination. Neurotransmitters, there's a lot here, and to be honest, I don't think we need to memorize them all. One of the things I would like you to have, though, is kind of the diversity. So you have everything from amino acids. That means amino acids are being used to make proteins, but they also can be neurotransmitters. And that's interesting when it comes to things like glutamates, because monosodium glutamate, or MSG, often gets a bad rap because people think it leads to migraines and hyperactivity and things like that, which on the one hand kind of makes sense because why would you eat a neurotransmitter that's a positive neurotransmitter? But also glutamate's just being used to make proteins, so maybe it's just helping you make more amino acid proteins. And that's why they put it in food, is because we taste it as meat. MSG adds meat flavor to foods. You got acetylcholine. This one's interesting because it will work in muscle, but it also works in the autonomic nervous system. So one of the bigger things, again, don't memorize all this, just see the complexity of the different neurotransmitters. Is there some that directly bind to receptors? So we talked about ligand-gated channels. All the amino acids over here bind to ligand-gated channels. Haven't really talked about it, won't really talk about it too much until we get to the endocrine, but there's other types of receptors that will bind to the outside of a cell, binds to something called a G protein. The value of the G protein is once the signal reaches the outside of the cell, it's magnified inside the cell hundreds, millions of times over. So, for example, one receptor will bind to the outside of the cell, and that'll change 10 molecules on the inside. But each of those 10 molecules will then change another 10 molecules, and then those 10 molecules, now 100 molecules, will change another 10. So you got up to 1,000. So you get this huge rapid multiplication. And that's the way the neurotransmitters on the right. So there's amino acids, and then there's big peptides like substance P is known to cause increased pain, or endorphins reduce pain. You got things like ATP, of all things, seems to be a neurotransmitter. Nitric oxide, carbon dioxide, also carbon monoxide, all these things seem to be neurotransmitters. So it's an awful, awful complex thing. So again, don't memorize individual things. Just see the complexity and understand that we manipulate these a little bit with pharmacology. There's probably 50 or more neurotransmitters. Clearly, we haven't identified all of the neurotransmitters, but there's some neurotransmitters that are just kind of used repeatedly. So the chemical ones that we know are definites are acetylcholine, biogenic amines, amino acids, peptides, and then there's some novel ones. Acetylcholine, really interesting because it's the one that is used to contract muscle, but it's also a big part of the autonomic nervous system, which is your fight or flight nervous system. You got catecholamines and indolamines. Probably have heard about these. Maybe not as catecholamines, but dopamine is a real pleasure neurotransmitter. Norepinephrine and epinephrine are like adrenaline. Serotonin, a big mood neurotransmitter, also involved in sleep. And histamine, of all things, involved in inflammation. Synthesis of catecholamines, this is kind of an interesting sequence because it plays a role in Parkinson's. What happens in Parkinson's is you have neurons that stop making dopamine. So it leads to inability to initiate movements and stop certain types of movements like shaking. So it starts with tyrosine, which is an amino acid. Tyrosine hydroxylase is the enzyme that stops functioning in Parkinson's disease. And that's why for a long, long time, as these neurons die, you can give the person L-DOPA and it will reestablish the amount of dopamine. Also really interesting that you start with an amino acid and you get three really important neurotransmitters: dopamine, norepinephrine or noradrenaline, and epinephrine or adrenaline. So why do we need to know this again? You don't need to know all of this in detail, but just kind of understand that there's a lot of complexity and it might come up later depending on your area of expertise with regards to pharmacology. There's amino acid ones: GABA, primary inhibitory neurotransmitter in your body. Glycine is also inhibitory. Glutamate's very, very stimulatory. And aspartate binds to a type of receptor called an NMDA receptor. Peptides, substance P is interesting because after you hurt yourself or cut yourself, usually the area around that cut is more sensitive to pain. That's due to substance P. Interestingly, too, endorphins reduce pain. So these proteins can act as opiates, but they can also increase pain. Some of them are somatostatin, which are hormones that are involved in hunger, things like that. So ATP, yeah, that ATP, the ATP that you use to drive muscles and things like that, can also be a novel neurotransmitter. Nitric oxide only exists for a couple of milliseconds, but that might be a neurotransmitter. Carbon monoxide also might stimulate certain neurotransmitters. Remember again, where we bring these all back, there was usually neurotransmitters are considered excitatory or inhibitory, so they either generate IPSPs or EPSPs. Some neurotransmitters, that's interesting because they can do both. So acetylcholine, it stimulates muscles, but often it's inhibitory in the autonomic nervous system. For example, it slows down your heart. Kind of mentioned this, you can have direct or indirect neurotransmitters. So sometimes the neurotransmitter binds right to an ion channel and opens it. Sometimes it acts through second messengers like G proteins. So this is a direct. The neurotransmitter binds to the outside, opens up the channel. Indirect neurotransmitter binds to the outside, causes a change on the inside. Usually there's a huge amplification here, and these might actually go to the DNA and mess with things in the DNA, or it might go and open up other ion channels. So G protein receptors, often they cause a huge magnification. I just found this figure kind of interesting because it talks about Botox. What Botox does is what's shown here is this is the presynaptic neuron, And this is a synaptic vesicle that normally fuses here, and it's holding neurotransmitters. What Botox does is it cuts these proteins that hold the vesicle next to the plasma membrane. So if you inject this in the muscles in your forehead, you won't have any synaptic vesicles lined up to cause stimulation of the muscle, and so you'll paralyze the muscles. But this is also why Botox can be very, very lethal. Just some other drugs, just to show kind of some of the things. Spider venom stimulates neurotransmitter release, so you dump all the neurotransmitter at once. Nicotine mimics nicotinic acetylcholine receptors and sometimes cross-reacts with muscarinic, which are autonomic nervous system. SSRIs block the reuptake of serotonin. Cocaine does that too. You can give certain drugs to increase L-DOPA, and that's important in Parkinson's. Curare and atropine block receptors. You can give drugs that inhibit acetylcholinesterase, which is the enzyme that breaks down acetylcholine. So again, just lots of different drugs that can interact with neurotransmitters. Next up is methods of processing. Probably should have been 10 there. Won't go into a whole lot of detail, but we've kind of talked about how you have to stimulate the axon hillock. And you have a bunch of pros and cons out here that stimulate the axon hillock in various ways. The summation of all those things, kind of said that wrong, is summation. So the input of all those pros and cons is called summation. You can have no summation, so you didn't get enough to reach threshold. So maybe you had two different stimuli, but they're too far apart to actually cause a stimulation. This would be temporal summation. So maybe you've got two different inputs happening at the same time, and that leads you to threshold. Maybe you have spatial summation, so you have two different pros. Maybe you have IPSP, EPSP summation, where you've got different pros and cons weighing in on the axon hillock together. So the only point of this slide is to show that really it comes down to temporal summation. Do you have lots of pros happening at the same time? Maybe you've got spatial summation, so they're not arriving at the axon hillock at the same time, but you have lots of different pros interacting at the same time. So really not going into too much detail, just showing you that neurons do interact in a variety of different ways. So let me get to these. So like you can have divergent input. The way I think about this is once you've decided to take an umbrella, you've got to alert the brain to figure out where your umbrella is. You got to make room in your bag to figure out where I'm going to put it. You have to decide what umbrella. So one decision leads to many, many other decisions, and that's called divergent processing. You can have convergent processing. Maybe you got a lot of pros that act on one neuron, so they come together and really, really stimulate. So this is what I think about when I think about Ella. I don't know what it is, but when Ella is involved with the rain, there's a lot of input going into my do I take an umbrella neuron. There must be a lot of convergent stimulation. Well, she cried a lot. I don't like to see my daughter cry. I certainly don't like to hear loud noises, things like that. So a lot of neurons came together to say, don't let that happen again. These guys are called parallel after discharge, and what they do is they create huge bursts of impulses. So this is involved in complex mental processing. Serial processing is when one decision leads to another decision, leads to another decision. And so maybe that's one of those where you get up out of bed and you stub your toe, and then you can't find your toothbrush, and then you can't find your car keys. And so a lot of things line up to say, hey, I'm not going to class today. Parallel processing, a lot of decisions happen all at the same time. And so maybe this time you get up out of bed and you fall and you stub your toe and the dog is puking all over and a bunch of things are happening all at the same time to say, nope, not going to school today. Reverberating, what reverberating do are they're kind of feedback, small feedback, and what they're involved in is really rhythmic things like sleep-wake cycle and breathing. So that's methods of processing, and that's the umbrella metaphor for the most parts. Again, if you can come out to this one, I'll probably just cut and paste the video right in. Here's the whole umbrella metaphor. It's broken into 3 parts, so you'll see here, I don't know if I can scrub through this, but there's the simple version.
Is that each individual question, I can explain it well enough that you understand that little piece of the puzzle.
So this is a simple version, takes you through the initial major chapters or major steps. What are the initial conditions? Why do ions move? How do ions move? What happens when they move? And then takes you through that a decision is basically pros and cons. If we jump out a little bit further, I'll add to the drawing. So again, we've got those basic components, but I've added things like how does fast signaling occur? And also down over here, I've got Ohm's law. So I've got pros and cons and how do you get various levels or strengths of inputs. And then if you jump to right around here, Somewhere right around here is the complete version, so you've got everything drawn. So you've got the basic building blocks. What are the initial conditions? Why do ions move? How do ions move? What happens when ions move? Action potential. Got Ohm's law here to determine the size and the strength of different inputs. Got synaptic transmission, so I've got IPSPs and EPSPs to demonstrate pros versus cons. And then a little bit about synaptic transmission. And lastly, there's a little bit on neurotransmitters. So if you wanted to skip those first two introductory levels, you could jump to right about here in the movie and watch it till the end, and I'll take you through each step one time, giving you the whole story of the umbrella metaphor.
Our next major topic is going to be nervous tissue. And the way I like to teach nervous tissue is through a metaphor. And I call the metaphor the umbrella metaphor. The reason I call it the umbrella metaphor is because whether you're aware of it or not, each time you hear rain, your brain has to make the decision of whether you take an umbrella or not. And what I really want to get at is that's what neurons do, is they make decisions. But before we get into that in too much detail, I just wanted to give you the heads up of what the expectations are for this test. And basically the expectations are that you're going to be able to reproduce this whole drawing. Now this is a simpler drawing right now. I'm going to take you up through 3 layers. By the time we get to the 3rd layer, it's going to be a pretty complex drawing. The main philosophy here is that if you can put everything into a story, then you really kind of understand everything. If you look individually, I've got things kind of like chapters in a book. There's a question 1, a question 2, a question 3. And my hope is that each individual question, I can explain it well enough that you understand that little piece of the puzzle. But then it's going to kind of be on your shoulders to put those pieces of the puzzle together to see the big picture. And if you do make it through that step to seeing the big picture, then you basically understand everything about how neurons make decisions. It's going to be really hard for you to just memorize this without understanding things, and that's kind of the overall point. Now, if you think about it really briefly, you can understand how you make decisions. There's usually pros and cons. Those pros and cons might be of different strengths. But what we're trying to get down to is how the neuron makes that decision for you. The other reason we're doing this today is basically this is our first step in understanding how some of the major electrolytes play a huge role in overall health. So we're going to talk a lot about sodium and potassium, calcium, and chloride today. Those are going to be flying around all over this sheet of paper. And so those are major electrolytes that we talked about when we talked about chemistry, and I told you you needed to have a basic understanding of ions and things like that. And those are going to play a critical role in how neurons make decisions. And these ions are going to just keep popping up. They're going to play a role in muscle contraction. They're going to play a role in how fast the heart beats, how hard the heart beats. They're going to play huge roles in how the kidney filters. And so this is kind of our first step in really understanding that if sodium and potassium get off, a lot of things can go wrong in the body. Sodium and potassium hardly ever get off in the body, and the body is just so good at controlling their concentrations, usually through the kidneys. But if they do get off, it's really, really tragic. And examples that you might be able to think about are Terry Schiavo. She had a problem with potassium levels. Basically, it was tied to diet. Then also the water drinking contest would have diluted out all these ions. Would have disrupted neuron function, so the woman became demented. It also would have caused edema, would have caused brain swelling. Where we're going to start on this umbrella metaphor is down here, number 1. I don't know why exactly I put everything in this kind of order. I think mainly when I first started to draw this out, this cell model here at the bottom is going to be least important by the time we get to the end. So I think that's where we have to start, but it's at the bottom because it's the least important later on. So again, this circle here is basically a cell model. It can represent any cell in the whole entire body. It certainly represents this neuron here, maybe just the soma or the cell body of this neuron. But it's just a cell model, a model of any cell. And the first thing I want to go into is something called the sodium-potassium ATPase, because it sets up some initial conditions that are common in all cells. So what the sodium-potassium ATPase does, just by the word ATPase, means it's an enzyme that breaks down ATP. And when it does that, it moves sodium and potassium. So sometimes it's called the sodium-potassium pump as well. What it specifically does, and I've just got one drawn here, but there's going to be millions in the cell, is it pumps 3 sodiums outside the cell, and in exchange it puts 2 potassiums back in the cell, and it burns an ATP. So it pushes 3 sodiums out, 2 potassiums in, and burns an ATP. There's going to be 2 results of that. I think the first one's pretty clear that it's going to push all the sodium to the outside of the cell. So you notice I got a lot of sodium on the outside of the cell. And it's going to pump potassium inside the cell. So it's going to set up this chemical gradient, where sodium's on the outside and potassium's on the inside. Maybe a little bit more difficult to see, but if you pump 3 positive sodiums out and only 2 positive potassiums back in, you're essentially putting positive charge on the outside. Now the first guy that ever talked about this, he could have talked about this in 2 ways. He could have said the outside of the cell is positive relative to the inside, or he could have said the inside of the cell is negative relative to the outside. Now either one of those is true, and he just chose to talk about the inside of the cell relative to the outside. So we speak of the inside of the cell being negative in charge. We express charge in volts, and so in our case it's right around minus 70 millivolts. Just to make it a little bit more confusing, this is called voltage of the membrane. So Vm equals minus 70 MV. I just wanted to add too, just at the end here, that this setting up this gradient again is common in all cells. In neurons, we've kind of set up a battery here though, so we've got charge on the inside, and we can change that voltage. We can let this go up to positive voltages. Essentially that's an electrical current that neurons can use to carry electricity and make decisions really, really quickly. But again, all cells set up this gradient, and we kind of talked about this when we talked about active transport, that if you put all the sodium on the outside of the cell, it kind of acts as an exchange or a bartering mechanism where you can let the sodium come back in if it's going to bring something else like glucose back in with it. Our next chapter or question is, why do ions move? And it's fairly simple, but just wanted to point out really basically that if you allow sodium to move, maybe you make a channel or a hole in this cell so that sodium can move, where and why is it going to move? I think the first one's fairly obvious, that sodium is going to want to move in just by diffusion. Diffusion means that things don't like to be next to each other, like things don't like to be next to each other. So if sodium can, it wants to move inside the cell because there's not as much sodium there. So the sodium-potassium ATPase is going to set up a gradient, where sodium is going to move in because it wants to get away from other sodiums, and potassium wants to move out because it wants to get away from other potassiums. So they're going to spread by diffusion. Again, a little bit less easy to see is there's also an electrical gradient, where sodium is a positively charged ion. It would sure like to get inside the cell where the inside of the cell is negative. So just based on charge attraction, the positive wants to be next to negative. So sodium wants to move in by electrical gradient. Potassium has just the opposite desire. So potassium likes to stay inside of the cell because it's a positive charge and it's next to the negative charge. And so it's happy where it is. So then the question becomes, what happens if you open up a hole or open up a channel so that potassium can leave? It's going to leave because the chemical gradient is more powerful than the electrical gradient. So if you open up a channel and let potassium do whatever it wanted to, it's going to want to leave by the chemical gradient. The electrical gradient is going to hold it back a little bit, but the chemical gradient is going to win. Another little detail to point out too is if you could open up a channel that would let both sodium and potassium flow through it, sodium is going to flow in faster than potassium is going to leave. And the reason is because both the chemical gradient and the electrical gradient are making sodium want to come in. The chemical gradient is wanting potassium to leave, but the electrical gradient is wanting it to stay. So in the case of potassium, one force is pushing out, one force is drawing it back in. And that's going to come up in like the muscle. In muscle, when you open up an ion channel, it's an ion channel that will let sodium and potassium both flow through it. That doesn't happen very often. Most of the time, an ion channel is just going to let one type of ion flow through it. So there's a sodium ion channel, a potassium ion channel, but only rarely will the channel be a sodium and potassium ion channel. But again, in this particular sodium and potassium ion channel that's in muscle, sodium is going to come in faster than potassium will leave. Our next question up here, question 3, is how do ions move? So we've just established in question 2 that ions will move if they're allowed to. So then how would they be allowed to? And the answer is ion channels. There's 4 basic types of ion channels, and really the only 2 that we're really worried about at this point are voltage-gated and ligand-gated. But we're going to bring in mechano-gated and leak-gated just so we can talk about all the different ion channels, and also mechano-gated and leak channels will come up when we talk about senses. So let's begin here at the voltage-gated channel. The first thing I want to point out is a gate. So this right here is a gate that's going to keep the channel closed. And I got it kind of hard to see. It's hard to see, but it's kind of an L-shaped gate. And the other thing to point out are that there's these little positive charges along the gate. Now, if the inside of the cell, like we said before, is minus 70 millivolts, these positive charges on the gates are attracted to that minus 70 inside the cell. So this gate is going to slide in this direction, and that's the closed position. Now what if that voltage changed from minus 70 up to something like plus 30? Now I want to be clear at this point that it doesn't necessarily always have to jump up to plus 30. Sometimes it just jumps up to minus 55, just a small change in voltage. But I'm doing plus 30 just because it's more extreme. It's a little bit more helpful to demonstrate how this gate is going to open. But if it's plus 30, then these positive charges say I'm not really attracted to the inside of the cell anymore, and so the gate slides out. And that is what opens the channel. So the pluses are attracted to the minus 70, gate slides down and stays closed. Pluses are not attracted to the inside of the cell because now it's plus 30, the gate slides open. A little bit more simple is a ligand-gated channel. Basically a ligand-gated channel also has a gate that I'm showing right here. And what it does is it can bind a chemical. Here I've got a little chemical, maybe it's a neurotransmitter, that binds to that gate and opens up the gate so that sodium can flow in. If there's no chemical bound, like in the case over here, then the gate tips shut and sodium can't enter. Just quickly, because again, we won't really talk about these here, we'll talk about them later. Are mechano-gated? Mechano-gated, there's a similar gate like the voltage-gated channel, and it responds to force. The main example is going to be in the inner ear, and it will respond to the force of a sound wave. So a sound wave will push on this gate and open it up, or if there's no push, then the gate will close. There's also what are called leak channels, which I've tried to draw smaller here, and these are always open. Small little sodium channels that are always going to let sodium flow in, small little potassium channels that are always going to let potassium flow out. And again, these won't really come up until we get to special senses. The next little chapter is relatively easy, I hope. And I just want to point out what happens when ions do move. So if we go back down here to our model cell, just pointing out that sodium wants to move in. What's going to happen to this minus 70 millivolts if sodium is allowed to move in? Well, now you've got positive charge entering the cell, so that's going to make this minus 70 more positive. So when sodium is allowed to come into the cell, this voltage is going to change towards the positive. Depending on the type of cell, it might swing all the way up to positive 30. Now what would happen if we opened up a channel and let potassium leave? Remember, if you open up a channel, potassium will leave, and it's going to take its positive charge with it. When it takes that positive charge, it's again like pumping that positive outside the cell, which makes the inside of the cell more negative. So what you have here is a way to change this voltage back and forth, up to positive voltages and back down to negative voltages. If you open up a channel and let sodium come in, we go up to positive voltages. If you open up a potassium channel and let it leave, we go back down to negative voltages. So we can switch this back and forth. The only other thing I wanted to point out in this first version is what I have over here are the basis for making this decision. So again, neurons make decisions, and those decisions will have pros and cons, so positive and negatives, and those pros and cons can have various strengths. So our umbrella, our metaphor here is the umbrella metaphor because it's do I take an umbrella today? When I was developing this metaphor, it reminded me of my daughter who really, when she was around 2 years old, really hated the rain. And obviously I learned that the hard way because the first time I took her out, I was like, let's just be tough and we'll just go without an umbrella. We'll run and we'll be quick. Suffice it to say, this was one trial learning. It was the last time I ever did that again because it was not a good situation. So from then on, I knew that if I had Ella with me, I was going to take an umbrella. And you can look around this neuron, and you can look at different things that might weigh in on whether you take an umbrella. I mean, Most of the time, most people are like, I'm tough. I don't need to take an umbrella. That's going to be a con. I already have too much to carry, so I'm not going to take an umbrella. Maybe the only umbrella you can find is your Crayola or your Hello Kitty umbrella, and I'm not going to carry a Hello Kitty umbrella. I'd rather get soaking wet. Now, there's probably reasons why everyone would take an umbrella. Maybe if you're at a wedding. Maybe if you're at a funeral, not to be a downer, but that's the main example. That's probably the only time I'll use an umbrella these days. Maybe your mother-in-law tells you it's going to rain, and so that's going to be a really kind of a weak pro. Maybe you know it's the beginning of the semester, and if you're arriving a little bit late, the parking lot is going to be hugely full, and so you might take an umbrella then, knowing that you might have to walk a long ways. Or another example there, maybe you live in Iowa City, and there's just no parking in Iowa City, or at least when I was a student there was no parking, so you had to walk all the way to class. And so then I maybe took an umbrella. Or maybe your hair's just looking really fine today, and you're like, I got to take an umbrella today. So anyway, the main point here is that neurons have to put all these things together. Am I going to a wedding? Am I parking? And whether you're conscious of it or not, these kind of little factors are factoring into whether you take an umbrella as soon as you hear those rain droplets on your roof. Now, I don't want you to think that there's one neuron that's making this decision for you. It might be a group of neurons. Nobody really knows how this works. But at some level, there is a single neuron or a group of neurons putting together all these pros and cons to make the decision of whether you take an umbrella or not. All right, so that's it for the first version. We'll head into the second version now. Okay, so this is a second run-through of 3 run-throughs. And I'm going to try and make sure that each one of these run-throughs stand on their own. So I'm going to cover everything again that I covered in the first run-through. I'll probably be a little quicker on this second run-through, but it's good to hear things a second time. So again, we're doing an umbrella metaphor here. Basically, the main point is that neurons make decisions. And we all know that decisions involve pros and cons. And those pros and cons can be of various strengths. So what we want to know then is, how does the neuron apply pros and cons? And how do those pros and cons have different strengths so that the neurons can make our decisions for us? And where it all begins is in the model cell. And again, this is a model cell. It could be any cell in the whole entire body. It's a little bit more specific to neurons now because I'm adding voltage-gated ion channels to it. But essentially, all cells in the body are going to have something called a sodium-potassium ATPase. And that's going to set up some initial conditions in every cell. But these initial conditions are really important in neurons to allow them to use electrical signaling, which is really, really quick. So first of all, the sodium-potassium ATPase, or sodium-potassium pump, pumps 3 sodiums outside the cell for every 2 potassium it pumps inside the cell, and it burns an ATP. This creates 2 gradients. The first gradient is chemical, which simply means that if you pump sodium out, sodium's going to end up on the outside. And if you pump potassium in, potassium's going to end up on the inside, and that's a chemical gradient with sodium on the outside and potassium on the inside. A little bit more difficult to see, not a whole lot more difficult, but a little bit more difficult to see is that if you pump 3 sodium out, you're pumping 3 positive charges out. And if you're only pumping 2 potassium in, then you're pumping 2 positives in. So you're essentially pumping positive charges outside the cell. That's going to result in more positive charges being on the outside of the cell than inside. And the first guy basically got to decide if he wanted to say, is the outside of the cell positive relative to the inside, or is the inside negative relative to the outside? And he chose to talk about the inside of the cell. So we talk about how the inside of the cell is negative relative to the outside. And we express that charge in voltage. And it comes out to about minus 70 millivolts. This charge is called the voltage of the membrane, or VM, and VM is equal to minus 70 millivolts. So now we've set up these gradients where sodium is stacked up on the outside and potassium is stacked up on the inside. It's pretty obvious that these ions are not happy this way. They'd like to move. But there's actually 2 reasons why these ions would like to move. The first one is, like I said, just diffusion. Sodium would like to get away from other sodiums, so if it could go inside the cell, diffuse inside of the cell, that would make it happy, so to speak. If there was a channel that opened up and let potassium flow through it, then potassium's going to diffuse out just so the potassium can get away from other potassiums. And those are chemical gradients. There's also an electrical gradient, and that's set up by this minus 70 millivolts. Sodium is a positively charged ion. It's attracted to negative charge. And so it would be attracted to the inside of the cell and would like to come inside the cell. Potassium is positive, and it's kind of happy to be up next to this negative charge. And so it's following its electrical gradient already, and it would like to stay. What that means is sodium wants to come in for two reasons, the chemical gradient and the electrical gradient. Potassium wants to leave because of the chemical gradient, but it wants to stay because of the electrical gradient. But the chemical gradient is much more powerful. Therefore, potassium will leave if an ion channel opens. And the other more specific example that I mentioned in the first run-through is tied to certain sodium and potassium ion channels. That doesn't happen that often. Most of the time it's just a sodium ion channel or just a potassium ion channel. But if you had a sodium and potassium ion channel, which you do in muscle, then the sodium is going to enter much faster than the potassium is going to leave. The third thing we talked about is we've talking about how these ions want to move. Well, how would they move? And the answer was ion channels. There's four main types. There's voltage-gated, ligand-gated, mechano-gated, and leak-gated. And we're really only concerned with voltage-gated and ligand-gated at this point. You're going to see these pop up throughout this. We see some voltage-gated channels here, some voltage-gated channels here, and later you'll see some ligand-gated channels. Mechano-gated and leak channels come up later when we talk about special senses. Let's talk again about the mechanisms of these voltage-gated channels. Essentially, there's a gate in this voltage-gated channel, and that gate has positive charges on it. Those positive charges are attracted to the inside of the cell when the inside of the cell is minus 70 millivolts, and so it slides down and it keeps the channel closed. If for some reason the voltage would change up towards the positive, and again I wrote plus 30, but it could be as little as minus 55. Well, now that positive charge is not as attracted to the inside of the cell. The gate slides out a little bit and the channel is open so sodium can come in. That's how voltage-dependent channels open. Ligand-gated channels, another name for ligand is chemical, and it's usually a neurotransmitter in our case. If that chemical binds to the gates, it's going to open it and allow ions to flow. If there's no ligands, no chemical, then the gate won't remain open or won't open, and it's going to remain closed. Mechano-gated or mechanosensitive channels basically have the same kind of gate as a voltage-gated channel. It might actually be kind of a hair that sticks up out of the channel, and when force pushes on that hair, it opens up the channel. When there's no force, the channel stays closed. And usually, the main example of this is going to be sound-sensitive channels in the inner ear. When a sound wave hits the ion channel or hits the gate of the ion channel, it opens. If there is no sound and no force, then the channel stays closed. The last one is leak channels. Leak channels are really small channels that just remain open all the time. They might let sodium come into the cell, or they might let potassium go out of the cell. The next thing we're going to start to talk about are what are called action potentials. And before we get to that, I want to go back to the cell model and get a little bit more specific about the types of voltage channels. I guess I should have done number 4 up here first, which is just to point out that when sodium comes into a cell, it's bringing in sodium positive charge. That's going to make the inside of the cell more positive. If we opened up a channel to let potassium out, then that's going to make the cell more negative because a positive charge is leaving. So now let's get more specific about these voltage-gated channels. I want to start with the voltage-dependent sodium channel. First of all, it opens at minus 55 millivolts, so you only need a small change in voltage from minus 70 up to minus 55 to open this channel. The other thing about the sodium-dependent or the voltage-dependent sodium channel is it has an inactivation plug, a little plug that will swing in and close the channel. It's just got a small little timer on it that once the channel has been open for a while, it swings in and closes the channel. Voltage-dependent potassium channels don't have that plug, and they open at a different voltage. They open at plus 30 millivolts. So already, hopefully you're starting to see that there's a potential for a sequence here to change the voltage. If signaling, like we talked about before, involves changing this minus 70 up to plus 30 and then back down, we kind of have all the pieces to do that. If something causes us to get to minus 55, the sodium channel opens and sodium starts pouring into the cell. That sodium that's pouring into the cell is going to make the voltage go even more positive. And it's going to make it keep going positive until we get up to plus 30 millivolts. And when we get up to plus 30 millivolts, the potassium channel is going to open. The plug is going to go into the sodium channel, so it closes. Potassium is going to flow out, and bring our voltage back down negative. Now we can formalize these voltage changes in a graph like this, where we have the voltage of the membrane here, and those key voltages we've been talking about, minus 70, minus 55, plus 30, are all here on the y-axis. What we have here on the x-axis is time. And so we're going to track that when you're sitting there and it's not raining, your umbrella neuron is sitting in what's called the rest state. It's not doing anything. There's no reason for it to do anything because there's no rain. As soon as there's drops of rain on the roof, something is going to get us up to minus 55 millivolts. And we're going to talk about that something in a second. But something's going to get us up to minus 55 millivolts. When we get up to minus 55 millivolts, this voltage-dependent sodium channel is going to open and start letting sodium come in. So sodium is going to enter the cell, and like we said, when sodium comes in, it makes our voltage more positive, so our voltage starts to go up. This is called depolarization. When we get up to plus 30, so now we're at plus 30, the plug is going to come into the sodium channel, and the potassium channel is going to open. So potassium is going to leave the cell and take our voltage more negative. And there's a little bit of an undershoot or an after hyperpolarization, or it's just called a hyperpolarization, when the potassium channel stays open for just a little bit too long. And our voltage goes down from minus 70 to like minus 80. We recover through the sodium-potassium ATPase. It puts all the sodium back where it needs to be, all the potassium where it needs to be, and we get back to minus 70 millivolts, and we sit in rest mode again. Now there's a couple of things else to point out about this. There's something called refractory periods. There's an absolute refractory period and a relative refractory period. What an absolute refractory period means is you cannot get another action potential once you've already started an action potential. So there's no super duper action potential, there's no big action potentials. Action potentials are always the same size. And the reason they're always the same size is because once you've opened those sodium channels, they all open. There's no like opening a few more. There's none that lay and wait to see if you got a super duper signal. They're all open. So there's no such thing as raising the voltage past plus 30. You're already raising it as far as you can. So that's an absolute refractory period. You're in the middle of an action potential. You can't have another action potential in the middle of an action potential. There is something called a relative refractory period, and that begins after the voltage starts repolarizing. This phase is called repolarization. And the idea here is that after all the sodium channels have closed and the potassium channels are open, you can reverse things. You can get the action potential to go back up. But it's really, really hard to do. And the reason it's hard to do is if you think about it, all those potassium channels are opening. So potassium is leaving, making your voltage more negative. So even if you open up the sodium channels, it's kind of like trying to fill the bathtub up with the drain open. So sodium's coming in, making you more positive. Potassium's leaving, making you more negative. So a whole lot more sodium has to come in than potassium is leaving. And that can happen to reverse this, but it doesn't happen very often. So basically, if you want to reverse and get another signal right away after a first one, it's got to be a really, really, really strong signal. So those are action potentials and refractory periods. The next thing I want to come to is down over here. And I want to talk about this again, just the neuron model, because I want to talk about how what we're talking about here is a neuron that's trying to make a decision about, do I take an umbrella today? And I want to be clear that this is a metaphor. There may not be just one particular neuron that makes that decision. It might be a clump of neurons, it might be a whole bunch of neurons. But in the end, neurons make yes and no decisions. Do I take an umbrella? No, I don't take an umbrella. Do I turn my car a little bit to the left so I stay away from the side of the road? Do I turn it a little bit to the right so I stay away from the center of the road? Is that a T or is that an I? Is that the word the or is that the word them? These are all small little decisions that neurons have to make for you. And all of them involve pros and cons of various strengths. And so what I have here is just my umbrella neuron, a drawing of my umbrella neuron, which is, I mentioned already that Ella, when she was 2, she hated the rain, and this was one trial learning. I took her out one time in the rain without an umbrella, and I never did it again. So she's a very strong pro in influencing whether I take an umbrella. Maybe parking is a positive, maybe a wedding is a pretty strong thing, and then maybe my mother-in-law is kind of a weak pro, because maybe my mother-in-law says, DJ, it's supposed to rain, you better take your umbrella. Now I'm going to listen to that, but I'm probably not going to run over and grab my umbrella right away. But also, just as something looking forward, you have to keep in mind that maybe this thing can become a stronger pro. Maybe my mother-in-law has a really uncanny ability to know when it's going to hail or something like that. And she will say, well, it's going to rain today and it's going to hail. So this pro has to be able to learn to get stronger. There's also negatives or cons, like maybe the only umbrella I have is a Hello Kitty. That's true. I've never bought myself a nice male-like umbrella because I'm just going to be tough. I'm going to run, I'm going to get wet, it doesn't really matter, or I have too much to carry. So you get this idea that there's pros and cons, and there are various strengths. Now for the first part here, we're just going to talk about the pros and the various strengths. How do you get the various strengths? And it comes down to something called Ohm's law. Ohm's law is basically used mainly in electricity, and what it usually describes is electricity will flow faster if there's more of a driving force behind it and less of a resistance. But this relates to almost anything that flows, whether we're talking about water through a hose or sodium trying to get to sodium channels in what's called the axon hillock. It's all going to be determined by how fast that sodium flows. It's going to be determined by how much it wants to flow, what's the driving force behind it, and how much resistance is there. In order to describe this maybe a little bit more simply, I'm just going to say that there's something down here called the axon hillock, and there's a whole bunch of voltage-dependent sodium channels here, and they're waiting to get to minus 55. So if you're going to kick off something called an action potential, you've got to get these sodium channels activated. You've got to get them to minus 55. Now before I progress here, I want to take a step away and describe something called an action potential versus a graded potential. I've got a little cartoon down here just to show you what a graded potential is. In a graded potential, if sodium just comes into a channel and spreads around, that's a graded potential. So it's like sodium coming in and spreading around. And that's different than an action potential, where if you have a bunch of voltage-dependent sodium channels or voltage-dependent ion channels, when sodium comes in one, it's going to move to the next one and change the voltage of that one, cause that channel to open, and now more sodium is going to come in. And now the sodium is going to make this voltage change around this channel, so it's going to open and more sodium is going to come in. And you can see that it's kind of a stepwise change. So maybe it's the difference between one person throwing a bucket of water on a fire versus a chain gang, where you're basically passing a bucket of water repeatedly to put out a fire. These graded potentials occur out here, so these things are going to let sodium into a cell, but they're just going to basically spread out. They're not going to get any help propagating. But action potentials occur here. And just like I had voltage-dependent channels spread out here, I get voltage-dependent channels spread out here. So I'm going to have voltage-dependent sodium channels and potassium channels here. I'm going to have voltage-dependent potassium channels and sodium channels here. Same here and same here. So what can happen now is if sodium comes in here, it can go down the axon and stimulate the next channel. And then sodium will come in here, because that channel the voltage was raised, and it'll come over to this one. So now that sodium's going to make this channel say, hey, I'm not at minus 70 anymore, I'm at minus 55, so I'm going to open and let sodium come in. And you can see that you can step down the axon that way. So the key here is to get these first guys to open. So that's why we're keying on the axon hillock right now. Is if you can get the axon hillock to minus 55, sodium's going to come in and get down to this one to turn this ion channel on. Then sodium's going to come here and get down to this ion channel and get this ion channel to be turned on. Then the sodium's going to make it to this one, and you've got a signal that's going to propagate down a neuron. So if your brain has decided to wiggle your big toe, it can send the signal down to the end. And we're going to see in a little bit that the point is to get the end, the synapse here, to plus 30, because then that will initiate a signal from this neuron to whatever its target is. Okay, so long divergence there, but the point I was trying to get is you've got to get the axon hillock, these sodium channels in the axon hillock, activated in order to kick off the signal to do something. We're going to just be really metaphorical here, really simplistic, and say maybe you need 1,000 sodiums here to get these sodium channels opened. Well, these different pros out here are going to have various abilities to get 1,000 sodiums down to this axon hillock, and those abilities are defined by Ohm's law. And maybe it's just going to be best if we get started on it, because first of all, the first part of Ohm's law that determines the strength of input is if the synapse is closer to the axon hillock it's going to be a stronger synapse. So Ella here is really, really strong. If Ella is with me and it's raining, I'm going to take an umbrella. Maybe one of the reasons she's strong is because she's right next to the axon hillock and right next to those sodium channels. Mother-in-law is way out here. So when she says take the umbrella, all the sodium that she's going to put in to get me to 1,000 sodiums down here is way out here. It's got to go a long ways. So Ella is at an advantage. She's going to be stronger because she's closer to the axon hillock. The other thing that determines the strength of the input is the size of the dendrites. Maybe we'll key here on parking. Parking is on a little bit bigger of a dendrite here than mother-in-law. So if both of these, just disregarding how far they are from the axon hillock, but if both of these can put in the same amount of sodium, just the fact that this is a bigger dendrite means this parking is going to be a stronger synapse. Maybe another metaphor is if you're trying to move 1,000 people and you've got nice big halls to move them down, you're going to be able to move them easily. And so switching back to the metaphor, if you can move 1,000 sodium ions down this nice big hallway, mixing my metaphors, then it's going to be easier to get that 1,000 sodiums down here. Now if you're trying to move 1,000 people through really narrow hallways, they're going to get all spread out. It's going to be hard to keep them as a group. And again, mixing my metaphors, if you put 1,000 sodium in here, this narrow hallway, by the time they get down to the axon hillock, they're going to be so spread out that they're not going to have that much influence on the axon hillock. The third thing that goes into the strength of the synapse is basically how much neurotransmitter is released, or how much sodium can that synapse put into the target neuron. So maybe Ella, she puts in 20,000, 100,000 sodiums, so you can be darn sure that 1,000 sodiums are going to make it down here to the axon hillock. Maybe mother-in-law, she can only put in 100 sodiums, so she's really, really weak. She's going to maybe play a role if somebody else gets us up to 999. She can weigh in on a decision, but on her own, she's not strong enough to make me get 1,000 sodium ions down here to the axon hillock and trigger the response. The other thing I want to point out about this is learning. So one of the main things is after you make a decision, you want to learn from that decision so that it influences future decisions. And basically all three of these might play a role in learning, but some of them more than others. So first of all, the wider the dendrite, well, let's start with one. The closer the synapses to the axon hillock. There's some evidence, pretty weak evidence, that maybe, let's just talk about mother-in-law. Mother-in-law has an uncanny ability to recognize when it's going to be a real bad rainstorm. And so when mother-in-law, after 25 times my mother-in-law saying it's going to rain and I don't take my umbrella and then it really just downpours, I finally learned my lesson that when mother-in-law says it's going to rain, it's going to be quite a bit of rain, so you might want to take your umbrella. So how did this synapse become stronger? One thing it can do, although there's really weak evidence, is that it can grow a tangent that will move closer to the axon hillock. So it can grow over here, or maybe it can grow another synapse over here and move closer to the axon hillock. There's pretty good evidence that activity will determine the size of the dendrite. So if we start firing this axon, or if we start firing the synapse a whole bunch, the dendrite will respond by getting larger. It's like if you keep forcing 100 people through a hallway or 1,000 people through a narrow hallway, eventually something's going to happen to get that hallway to get bigger. So if you keep putting sodium in here, it will make that dendrite get bigger. So that's another way to learn. But probably the quickest way to learn is just to release more neurotransmitter. Mother-in-law has convinced me that she knows when I should take an umbrella, so this synapse is just going to release more neurotransmitter. So maybe the first 25 times mother-in-law says it's going to rain, I release 100 sodiums. It doesn't have any effect on the axon hillock. Maybe I finally learned my lesson, and now mother-in-law is going to release 100,000, a million sodium ions just by releasing more neurotransmitter. So there's definitely going to be enough sodium that gets down here to get my pretend or metaphorical 1,000 sodiums down at my axon hillock. So just trying to point out how learning can basically occur then. I think that's it for the second run through. We're going to come back through the run through in the third time and talk about synaptic transmission and how you have the negative effects. So this is the third run through of our umbrella metaphor, and I basically want this run through to stand on its own. So I'm going to repeat everything in this run through that I've talked about in the first two run throughs, so that this one will be the complete story that you'll need to study. I want to start by talking about what you do need to study and what's going to be expected of you on the test. And basically, you'll be asked to reproduce this whole drawing on the test. You'll get a blank 11 by 17 sheet of paper, and you'll redraw this in full, and I'll check it against a list of everything that's on this drawing right now. The pedagogical argument here is that if you can put everything together into a story, it's not going to be that hard to reproduce this drawing. It will be awfully difficult to simply memorize this drawing if you don't understand things. Again, the philosophy is to try and present each individual part of this story really, really simply. So this can be viewed as like chapter 1 of a book, chapter 2 of a book, chapter 3 of a book. Hopefully those are fairly simple little parts of the story. And then it's basically on your shoulders to put this all together so that it all makes sense. And then once it makes sense, it's going to be really easy to reproduce it. The main goal here, or what we're mainly trying to understand here, is how nervous tissue makes decisions, or how neurons make decisions. With some pretty basic thought, it's really easy to understand how you make decisions, how your decisions involve pros and cons, and those pros and cons can be of various strength. And these decisions can be everything from, like our metaphor today, do I take an umbrella, to is that a T or an I? Is that the word the or them? Is my car moving to the left and so I need to steer a little bit to the right to stay in my lane? Is that something off to the side of the road that I need to respond to? These are all small little decisions that guide our overall consciousness. But if we go back to the neural level, basically what neurons do is they gather a lot of input, pro and con input, and they come to a yes or no decision. Another really critical subtext is we're starting to begin to look at electrolytes, really important electrolytes like sodium, potassium, calcium, and chloride. Now, we kind of touched on this back when we talked about chemistry, and we talked about ion bonds, and we talked about how sodium and chloride will dissociate in water, and they'll kind of relate to each other, and they have a charge reaction and things like that. And so we're going to build on that now. And it's going to be really an important build because sodium, potassium, calcium, and chloride are going to come up continuously throughout AMP1 and AMP2. It's one of the last things we talk about in AMP2 when we talk about the urinary system and electrolyte balance. We're going to talk about when we talk about how muscles contract. We're going to talk about with the endocrine system because calcium has a role there. We're going to talk about with the reproductive system because calcium has a role there. We're going to talk about it and how it sets the heart rate because sodium and potassium set the heart rate. We're going to talk about how muscle contracts in the heart as well. So anyway, these ions, these electrolytes are going to keep popping up over and over again. And one of the biggest things I want you to take away from AMP1 and AMP2 is respect for these electrolytes and this electrolyte balance. One of the hard parts of developing that respect is the body is so good at regulating these ions, and they so rarely get off. But if they do get off, if there's an imbalance in these ions, it's almost always immediately tragic. Some examples that you can think about are Terry Schiavo, due to diet problems, had an imbalance of potassium that shut down her heart and led to a coma. Or another really kind of unfortunately infamous event happened, I think, last February with the water drinking contest. If you drink too much water, it causes water toxicity, which basically is relating to these electrolytes because you're diluting out these electrolytes. And as you'll see here today, that's going to affect how neurons function. So beginning again at the bottom of this sheet with the model cell, this is essentially a model cell. It's got a few things added to it now because we're in our third run through, but at its heart, you have a sodium potassium ATPase and you have a plasma membrane, and you're distributing sodium and potassium due to that sodium potassium ATPase. And again, this model cell will represent any cell in the body, and this certainly is going to represent this soma right here. This is the main thing that we're going to focus on right here, is we can pretend that this sodium potassium ATPase down here is actually in the membrane of this neuron. So let's talk about this sodium potassium ATPase first. First of all, ATPase means it's an enzyme that breaks down ATP. And what it does is it moves sodium and potassium across the plasma membrane. Specifically what it does is it pumps sodium. So a lot of the time, this is called the sodium-potassium pump. And again, specifically what it does is it pumps 3 sodiums out and 2 potassiums into the cell. It's going to set up 2 gradients. The first one hopefully is fairly clear, that if you're pumping sodium out, you're going to end up with more sodium on the outside than on the inside. And if you're pumping potassium in, you're going to end up with more potassium on the inside than on the outside. The other thing might be a little bit harder to see is it's going to set up an electrical gradient too, because you're pumping 3 sodium ions out or 3 positive charges out, and you're only pumping 2 potassium ions in or 2 positive charges in. So essentially what you're doing is you're pumping positive charge outside the cell. Now the first guy that talked about this, he had a choice. He could talk about the outside of the cell is positive relative to the inside, Or, same thing, he could say the inside is negative relative to the outside. And he chose to speak about the inside of the cell as being negative relative to the outside. More specifically, it's about minus 70 millivolts. Millivolts, or volts, is the unit of charge. We can also speak of this as the voltage of the membrane, which is abbreviated as Vm. So Vm equals minus 70 millivolts, or MV. So now the sodium-potassium ATPase has set up these initial conditions, and hopefully we can see that ions want to move. But let's get more specific about why do ions want to move, and that's number 2 here. Again, it comes down to those two gradients set up by the sodium-potassium ATPase, the chemical gradient and the electrical gradient. The chemical gradient is simply that sodium is stacked up on the outside of the cell, and it would like to move away from itself. One sodium ion would like to move away from other sodium ions just by diffusion. Potassium has the same situation. It's stacked up on the inside of the cell. Each potassium ion would like to get away from other potassium ions by diffusion, so it wants to move outside the cell. There's also an electrical gradient set up by that minus 70 millivolts inside the cell. And sodium and potassium are going to react differently to that minus 70 millivolts. Because sodium is on the outside, it's positively charged, and it wants to come into that negative charge because opposites attract. So the positive sodium wants to be next to that negative charge. So sodium is going to be drawn into the cell by the electrical gradient. Potassium is already next to that minus 70, so it really doesn't want to leave if you look at just the electrical gradient. Potassium does not want to go against the electrical gradient. Now if you put the chemical gradient and the electrical gradient together, the chemical gradient is going to be much stronger than the electrical gradient, and so potassium is going to leave. Another way we can think about that is if you could open up a channel that would let both sodium and potassium through, what would happen? Now this is a rare situation because most of the time an ion channel is just going to let one type of ion through. So there's a sodium channel that lets sodium through, or there's a potassium channel that just lets potassium through. There is one specific case that is going to come up when we talk about muscle physiology, where a channel will let both sodium and potassium flow through the same opening. And in this case, sodium is going to come in faster than potassium leaves. And that's because sodium again is being drawn in by both the chemical gradient and the electrical gradient, while potassium wants to leave by the chemical gradient, but it wants to stay by the electrical gradient. So hopefully it's clear that ions want to move. The next question then, question number 3, becomes how do those ions move? And the answer is ion channels. And there's 4 main types of ion channels. There's voltage-gated channels that are going to respond to the voltage of the membrane, or VM. There's ligand-gated channels that are going to open when a chemical binds to them. There's mechano-gated channels, and they're responding to force. And the main example here is going to be a sound wave. And then there's leak channels that are really, really small, but they're open all the time. So I'm focusing in a little bit more, and I'm going to start on the voltage-gated channels so that we can describe why those open and close. We're going to start with something called a gate, and obviously these are real simple cartoons. And right here is our gate, and that's going to keep the channel closed. On the gate are positive charges in the case of the voltage-gated channel. These positive charges are going to respond to the voltage of the membrane inside the cell. If the inside of the cell is like minus 70 millivolts, those positive charges are going to be attracted to the negativity inside the cell, and so this gate is going to stay slid down. If we change the voltage towards positive, now here I've got it as plus 30 millivolts, but in the case of a sodium channel, it might be just a small shift in voltage up to minus 55 millivolts. But if you move a little bit positive, then those positive charges say, I'm not really attracted to the inside of the cell as much now, so I'm going to slide out. And when it slides out, the channel is open and an ion can pass through. Ligand-gated channels are considerably simpler. Basically, a ligand is a chemical that when it binds to the gates, it opens the gates. If there's no chemical bound, the gate stays closed and no ions can pass. Again, mechano-gated are going to respond to force, and in the inner ear, you have these types of channels with small little hairs sticking out. And when sound waves come along and push on those hairs, it can open the channel. If there's no sound wave to push on the hair, then the channel stays closed. Leak channels are really, really small channels that just let sodium flow in all the time and potassium flow all the time. To be honest, for the most part, we're really only concerned with voltage-gated and ligand-gated channels at this time. And you're going to see those popping up. You'll see them on this figure here, that voltage-gated channels are drawn here. Ligand-gated channels are drawn over here. So these are going to pop up throughout the umbrella metaphor. Mechano-gated and leak channels aren't really in this lecture, but we're going to need to have an understanding of those when we do special senses, so that's why we're covering them here. Our next question is what happens when ions do move, and the answer is that the voltage of the membrane changes. I'll take you down here to the cell model, basically point out that what's going to happen if sodium does come into the cell. Sodium's positively charged, and if it's allowed inside the cell, it's going to bring that positive charge. It's going to make that voltage of the membrane go more positive. A little bit harder to see is if potassium leaves, it's going to take its positive with it. It's going to make the outside of the cell more positive, which means the inside of the cell is going to be more negative. So when potassium leaves, it makes the inside of the cell more negative. So what we have here is the opportunity to change this voltage up to positive voltages and then back down to negative voltages. And this ability to change voltage by letting sodium come in and raise your voltage and letting potassium leave to bring your voltage back down is the basis of cellular signaling. It's about as fast as a signal as you can create. It's not as fast as electrons, but these ions move pretty fast. And if you want to get some signal from one end of a neuron to another end of a neuron, changing the voltage is a pretty quick way to do it. Before we get into detail about these voltage changes and how you graph it out, I want to just get into a little bit more detail. So focusing back on this model cell, I just wanted to point out some details of the different types of voltage-dependent channels. We have two different types of voltage-dependent channels. I mean, all over the body you have different voltage-dependent channels. You have voltage-dependent calcium, you have different types of voltage-dependent sodium channels, and different types of voltage-dependent potassium channels. And that's going to be important, especially when we get to cardiac function. But right now we're going to talk about two types of channels, a voltage-dependent sodium channel and a voltage-dependent potassium channel. And they're very different as far as when they open. First of all, the voltage-dependent sodium channel is going to open at minus 55 millivolts. So to review, there's this positively charged gate that when the inside of the cell is minus 70 millivolt, this gate is going to be attracted to the inside of the cell and it's going to slide down and be closed. But if we move as little as 15 millivolts up to minus 55 millivolts, this gate is going to slide open and the channel is going to be open to let sodium come in. Kind of a unique feature of the voltage-dependent sodium channel is something called an inactivation plug, and this is going to swing in shortly after the channel is open and basically close all the sodium channels down. The voltage-dependent potassium channel is going to open at plus 30 millivolts. And when it gets to plus 30 millivolts, potassium is going to leave. So hopefully you've started to see the basis here of changing the voltage. Something's going to happen to get us from minus 70 millivolts up to minus 55 millivolts. Sodium channels are going to open, and sodium's going to start coming in. When that sodium comes in, it's going to raise the voltage even further. It's going to raise it all the way up to plus 30 millivolts. At that point, the inactivation plug is going to swing in and close the sodium channels, and the potassium channels are going to open. When the potassium channels are open, potassium is going to leave and bring our voltage back down to negative. So you have the mechanism here. Open up the sodium channel, get my voltage up to positive values, close that sodium channel, now open up the potassium channel and get my voltage back down. Now we can formalize these voltage changes by graphing them. What we're going to do is we're going to graph membrane potential here, and some of the key membrane voltages are minus 70, minus 55, and plus 30. We're going to graph the changes in these voltages over time. And what we get is a figure that is called an action potential. Basically, we're going to start right here at rest, and this would be the situation when there's no rain right now. My umbrella neuron is not doing anything right now. It's at rest. Now, if something happens, you hear some raindrops on the roof or something like that. Something, and we're going to leave it as something right now, but we're going to clarify it in a little bit. Something's going to get my voltage-dependent sodium channels up to minus 55. Now, when they open, they're all going to open, and they're going to bring the voltage up to plus 30. So that's why I have this figure here. The sodium channel is open and the potassium channel is closed. And this state is called depolarization. When we get to the top here, the inactivation plug is going to swing in. Hopefully you can see that right there. And the sodium channels are going to close. The potassium channels are going to open. And this is going to do something called repolarization, which means it's going to take our voltage back down negative. The potassium channels stay open a little too long, and that causes an undershoot, or it's also called a hyperpolarization. The sodium-potassium ATPase gets everything back in order, so it gets the sodium back out, potassium back in, and that brings our voltage back to minus 70. Something else to mention, though, are refractory periods. There's refractory periods in the action potential, and one is called an absolute refractory period, and one is called a relative refractory period. What refractory period means is it's hard to get another action potential. And that can be absolutely hard to get another action potential. And that's what the absolute refractory period is. You cannot get another action potential if you're already starting up on an action potential. And basically what this comes down to is there's no super duper action potentials. There's one size of action potential no matter where you go in the body. You don't get big action potentials and small action potentials. The way you actually code for something that's a simple touch or a really hard touch or something that's really painful, basically the way you code for intensity is how fast the action potentials come, not how big they are. Now again, the reason that there is no super duper action potential is because once the sodium channels open, they all open. There's none that lay in wait in case there's a bigger signal. They're all open. And so if they're all open, you can't open any more than all of them, and so you're always going to have the same signal. So once you've started up on an action potential, all the sodium channels are open, so you can't get a bigger action potential, or you can't get another action potential in the middle of an action potential that's already occurring. Now once those sodium channels start to close and the potassium channels start to open, technically you can get another action potential, but it's really, really hard. That's why it's still called a refractory period. And the reason it's hard is mainly because those potassium channels are open. If we go down here to the model cell and we think about, well, to get our voltage back up, we have to have sodium coming in. But if the potassium channels are open and potassium is leaving, then it's a bit like trying to fill up the bathtub when the drain is open. You can do it, but you have to make sure that the water is coming into the tub faster than the water is leaving. So in this case, back to the cell, the sodium has to really, really be coming in. It has to be coming in faster than normal because now it has to offset the fact that the potassium is leaving. So you can get another action potential once you've started down, but it has to be a super strong signal. And these are reserved for cases when you really, really need a lot of action potentials in a row. And if we go back to talking about how you code for intensity by the frequency of the action potentials, this means it's really hard to stack up the action potentials really, really close to give a really, really intense signal. But if you do, now you do have that super duper signal, because not only have you stacked the action potentials close, but you've stacked them so close that more sodium is coming in than potassium is leaving. So you got that extra turn the dial up to 11 signal when you actually have such an intensity that the action potentials are right up next to each other and impeding on that relative refractory period. Now let's go down here to this lower left where I'm going to describe the two types of potentials. And this is where you really begin to understand what an action potential is. What an action potential is it's a potential that can move. And that's set up by the fact that you spread out voltage-dependent channels. And in this little cartoon, I've got a voltage-dependent sodium channel with sodium coming in. And if sodium comes in through that channel, it's going to spread to the next channel. And now that channel and the gate in that channel is going to say, I'm at a more positive voltage on the inside, so the gate's going to open, and now this channel is going to start letting sodium come in. Sodium's going to migrate over to this channel. This gate is going to sense that positive sodium on the inside, say the membrane voltage is not minus 70 anymore, it's minus 55, so now that one's going to open. And you get this jumping from voltage-dependent channel to voltage-dependent channel, and that's really what an action potential is. And where that occurs in a neuron is right along the axon. So you have myelin in here, you have gaps in the myelin that are called nodes of Ranvier, and in those nodes of Ranvier there's voltage-dependent sodium and potassium channels. So again, if sodium can come in here, this area is called the axon hillock. If sodium comes in here, it spreads, and it spreads to the next node of Ranvier where there's a voltage-dependent sodium channel waiting. Sodium comes in, and it spreads to the next node of Ranvier where there's a voltage-dependent sodium channel waiting. That sodium channel says it's not minus 70 anymore, it's minus 55, so I'm going to open and I'm going to let sodium come in. That sodium technically spreads in both directions, but the important direction is in this direction, because now this sodium channel is going to sense that voltage change and it's going to open. I'm also going to bring back that inactivation plug, which we can look at in the cell model. Basically, that inactivation not only closes the sodium channel, but it keeps it closed, and it makes sure that the action potential travels in only one direction. So bringing in two things there, don't mean to be confusing. Bringing in the action potential, which means things will jump down the axon in what's called saltatory conduction. And also, the reason we have that inactivation plug is it prevents the action potential from backing up the axon, basically going upstream. Because even though the sodium might flow back to the previous node of Ranvier, the sodium channels in that node of Ranvier are going to be inactivated. And again, this keeps the action potential going in one direction. The other type of potential is just a graded potential. In a graded potential, there aren't voltage-dependent channels to help propagate the signal. So when the sodium comes in, it just spreads around. Graded potentials occur out here. So at this point, we've kind of hit on the fact that this axon hillock is really important, because this is where there's a huge cluster of voltage-dependent sodium channels that are trying to get to minus 55 millivolts. And really, the whole decision about whether I take an umbrella or not is made right here at the axon hillock. What it's going to do is weigh pros and cons. So it's going to weigh information from pro neurons out here, pro synapses, and negative synapses, and decide, do I fire this axon? How it decides that is basically, do I get enough sodium to get up to minus 55 millivolts to open these sodium channels? Because if they do get opened, then sodium's going to flow in. You're going to send your signal down the axon, and you're going to signal to another cell. This umbrella thing is a go, so start planning it, or squeeze the muscles in the hand to pick it up. But we are going to take an umbrella. Things like that. So then the question becomes, how much sodium does it take to get these sodium channels open? And we're going to just put a fake number on it. We're just going to say 1,000 sodium ions. Now it's going to be way higher than that, but keeping it simple, like something like 1,000, is going to help us focus on what's going to make us get to 1,000, like Ella, and what's going to keep us from getting to 1,000, like I want to look tough. But for this first part, we're just going to talk about the pros. What are the things that are going to try and stimulate the axon hillock? Because there's 3 things that go into how strong Ella's effect is on the axon hillock, how strong the mother-in-law's effect is on the axon hillock, how strong my hair is looking fine today is on the axon hillock. And these strengths of input are defined by something called Ohm's law. Now, Ohm's law is usually described in terms of electricity. It's like how fast current flows is dependent on how much it's pushed by the voltage over how much resistance there is. But Ohm's law will describe a lot of things. It describes water through a hose too. How fast the water comes out of a hose depends on how much pressure is on the hose behind it and what resistances are on that water. So if you have a big hose, you have less resistance and more water is going to flow. Things like that. Ohm's law also describes the likelihood that Ella is going to get 1,000 sodiums down here to the axon hillock. It's going to describe the probability that mother-in-law is going to get 1,000 sodiums down to this axon hillock. The first thing that's going to describe whether a synapse has the intensity or the strength to stimulate the axon hillock is how close is it to the axon hillock. So I kind of imagine in my brain the Ella synapse is really, really strong, and one of the ways it can be really, really strong is it's right next to the axon hillock. So if it dumps 1,500 sodium ions in, chances are 1,000 of them are going to make it to the axon hillock, just because that synapse is so close to the axon hillock. Mother-in-law, on the other hand, whatever sodium she dumps in here, it could go in a variety of different directions. It's going to get spread out by the time it gets to the axon hillock. So the fact that this synapse is so far from the axon hillock is one of the reasons this synapse is relatively weak. Another thing described by Ohm's law is the width of the dendrite. And basically a metaphor here is if you wanted to move a bunch of people, would you want wide hallways or would you want narrow hallways? With wide hallways, you could move those people and you could keep them right together. If you have narrow hallways, they're going to get spread out. So to kick off the axon hillock, we need 1,000 sodiums in a clump to get down here. So if we've got a nice wide dendrite, or in this case on this parking dendrite, a nice wide dendrite, it's going to be easy to move the sodium in a clump down to the axon hillock and make sure we get 1,000 sodiums down here to stimulate the axon hillock. If you're way out here on mother-in-law, really, no matter how much sodium you dump in there, it's going to get spread out. And so it means this synapse is going to be very, very weak. It's going to be hard for this synapse to get 1,000 sodiums down here. The third thing is simply how much neurotransmitter is released. So when I took her that first time, and it was a bad situation, I needed to learn once. I needed to learn really quickly. And probably the easiest way for a neuron to improve its performance is to release more neurotransmitter so more sodium is released into the target neuron. So if Ella releases 500,000 sodium, it's pretty likely that 1,000 sodium are going to stimulate the axon hillock. And kind of extending on that, let's say, for example, let's extend into learning. Let's say, for example, that mother-in-law has this uncanny ability to predict really, really strong thunderstorms. And so for the first 25 times, my mother-in-law says, DJ, it's going to rain today, you should take an umbrella. I blow her off, and I get really, really soaked in a downpour. Eventually, I'm going to want to learn to make this synapse stronger. It's very weak right now because it's so far away. It's on a very narrow dendrite, and it's not releasing a lot of neurotransmitter. So how is it going to become stronger? Each of these methods can lead to improvement, but some are going to be easier than others. For example, a really weak one, but it might actually occur, is this synapse might grow an additional synapse off to the side that's going to be closer to the axon hillock. And that does happen sometimes. Those are called collaterals. There is research that if a synapse is used often, the dendrite will get wider. And so if this synapse is releasing a lot of neurotransmitter, releasing a lot of sodium into here, the dendrite will actually grow, and that will improve this synapse's performance. Again, a nice wide dendrite means that the sodium can move in a clump and make it down to the axon hillock. But probably the most likely, and I already alluded to it, the most likely way that you're going to learn is you're going to have the synapse release more neurotransmitter. So mother-in-law, if she releases 1,000,000 sodiums, it's likely that 1,000 sodiums are going to get down here to activate the axon. So that's a look at how you can get different strengths of input, but it's also important to keep in mind that all these things kind of add together. So all the pros will add together to get 1,000. So maybe Ella's not with me. That's not going to get me my 1,000. But maybe parking plus mother-in-law plus the hair looks fine. Those three together are going to put in enough sodium that I get to 1,000 sodiums at the axon hillock. So just to come back to it and say it for about the 25th time, is neurons make decisions. Decisions involve pros and cons, and those pros and cons can be of various strength. And we've kind of already covered now how you can get pros and how they can be of various strengths, but we haven't talked about cons. And so to talk about cons, how do you negatively influence taking an umbrella? How does I'm tough shut this I'm taking an umbrella neuron down? First of all, I'm going to go over to the model cell and point out that I've added another ion, the chloride ion. Now, I didn't put the chloride ion in the first and the second versions just because we didn't need it up to then and I didn't want to add the clutter, but we definitely need it now, so now it is in the third version of the umbrella metaphor. Please notice that chloride is negatively charged and it's more concentrated on the outside of the cell. So one of the ways you can negatively influence the axon hillock, prevent it from getting to minus 55, is make the cell more negative. We can see that in a couple places. If you have to get to minus 55 and you're at minus 80, it's going to be harder to get up to minus 55. How do I get to minus 80? The simplest way is to let these negative chlorides in. If a negative chloride comes in, it's going to make you more negative. You could also open up a potassium channel, and when the potassium leaves by diffusion, it's also going to make the cell more negative. So everything up here under cons were in red. Notice that a con neuron is going to either let chloride in the umbrella neuron or it's going to let potassium out. Now these things, because it's changing the potential, are called inhibitory postsynaptic potentials. So the postsynaptic neuron, here's the presynaptic neuron, is the red neurons coming in or the green neurons coming in, and the umbrella neuron is what's called the postsynaptic neuron. So that's where the inhibitory postsynaptic potential comes from. Again, presynaptic neuron and postsynaptic neuron. While changing the membrane voltage to more negative voltages is probably the easiest to understand way that an IPSP has its function, there is another way that it works, and it's called a shunt. Basically what happens is these channels can just simply sit open. Then every time a sodium comes in, these channels will allow a potassium to leave or allow a chloride to leave. So then what's happening is every time a sodium comes in, it wants to bring in its positive charge, but it's offset by the negative charge of a chloride coming into, or offset by the negative charge of a potassium leaving. So it's kind of having a negative effect. Just like back to the bathtub metaphor, you can inhibit filling the bathtub by opening the drain. In this case, sodium would be pouring into the bathtub, mixing metaphors, but potassium and sodium would be leaving through the drain, and it would offset each other. So this is a technical, kind of a little bit hyper-technical way that these actually probably inhibit. On the pro side, it's much more simpler, because really the only way to change this voltage more positive is to let more sodium come in. And now that's called an excitatory postsynaptic potential, because it's going to excite the postsynaptic cell. In our case, it's this yellow, do I take an umbrella today, neuron. So then here, now that we've finished up with cons, take a moment to think about how there's kind of a quasi-math that's occurring in the postsynaptic neuron. Sodium's coming in from some pro over here. More sodium is coming in from this pro over here. Negative chloride is coming in from this con over here. So this mash of sodium coming in, chloride coming in, potassium leaving, that does a certain amount of mix-up math to determine, do you get those 1,000 sodiums down to the axon hillock? So this is why when we make decisions, we weigh a lot of pros and cons. The umbrella neuron is weighing a lot of IPSPs and EPSPs to determine, do you have enough sodium coming in to stimulate the voltage-dependent sodium channels in the axon hillock? If you do, then it's a yes decision, and if you don't, then it's a no. The next thing to talk about then is synaptic transmission. So if you've got these chloride channels opening, these potassium channels opening in the postsynaptic neuron, or these sodium channels opening up in the postsynaptic neuron, how did that signal get to them to open up? And it's really a fairly simple process we're going to outline here. Basically, on your drawing in class, you can either draw the figures or you can write it out. It all begins at getting to plus 30. You'll notice down here I've got plus 30. That's a key number. So when this action potential goes down, we make the presynaptic neuron plus 30. When the presynaptic neuron gets to plus 30, now there's a voltage-dependent calcium channel. So it's almost the same as the potassium channel and the sodium channel as far as it's got a small little gate that slides out when the channel gets to plus 30, and calcium can come in. When calcium comes in, it opens up neurotransmitter vesicles. You notice these small little circles are filled with black dots. Those black dots represent neurotransmitter, and the little circles represent something called synaptic vesicles. When calcium binds to the synaptic vesicles, they open up. Normally they're pinched closed, and they can just open a small little bit, and they're going to release their neurotransmitter, and they're going to bind to ligand-gated channels, the same kind of channel we talked about over here, ligand-gated channels. When the neurotransmitter binds to those ligand-gated channels, they open up, and in this case they let sodium in. Over here in the case of the cons and the IPSPs, they let chloride come in or potassium leave. Now there's one more critical thing, and there's kind of a rule of neurotransmitter release, which is you should release enough neurotransmitter to stimulate every postsynaptic receptor once and only once. So every receptor gets one neurotransmitter molecule, and then you have to get rid of the rest while those stimulated receptors are doing what they do. There's 3 ways that you get rid of the rest. And it's important to understand these processes because we have pharmacological interventions. We can give drugs that affect these processes. So just a little bit more closer of a look. The first process I'll talk about is something called reuptake. And so after we release all that extra neurotransmitter, one way we can get rid of it is to take it back up into the presynaptic terminal, and that's called reuptake. And one of the reasons this is important is because this is what SSRIs, selective serotonin reuptake inhibitors, or antidepressants do. They inhibit this process, and they leave the serotonin in the cleft longer, constantly stimulating the postsynaptic receptors. This effect of the drug takes place almost immediately, within hours of taking the drug. But maybe most of you know that antidepressants generally don't start working for 2-3 weeks at a minimum after taking the drug. So it's clearly not leaving the serotonin in the cleft that has the antidepressant effect. It's thought that that serotonin left there causes the body to say to these serotonin receptors, I'm being stimulated too much, so I'm going to take some of those away. And it's the taking away of those serotonin receptors that has the antidepressant effect, and that process is what takes 2 to 3 weeks. Another mechanism in different synapses is enzymatic breakdown. So I've got this small little circle which is supposed to represent an enzyme, and it can grab onto any neurotransmitter that hasn't found a receptor and basically break it down. This is going to occur mainly in the synapse between a motor neuron and a muscle. And sometimes there's a disorder called myasthenia gravis, where that receptor that's supposed to pick up the neurotransmitter gets eaten away at by the body's own immune system. And one way you can give a drug to fight that is to inhibit the enzyme. If you inhibit the enzyme, the neurotransmitter sticks around longer. So even though there's less receptor, the muscle still gets stimulated, and you don't get paralyzed as fast, or the muscle weakness is attenuated a little bit. The third mechanism of getting rid of excess neurotransmitter is some of it just diffuses out of the synaptic cleft area. The last thing to talk about are what are the different types of neurotransmitters, and this is an oversimplification to be sure. But in general, we can divide neurotransmitters up into direct type neurotransmitters, and these are going to be the type that bind to ligand channels and open up those channels. You can also have indirect ion channels, and these are going to bind to G proteins, something we touched on early in this class. Basically what a G protein does is it binds something to the outside, and then through a cascade effect, it multiplies that signal up millions and millions of times, so you can change a whole host of proteins or a whole host of processes inside the cell. Direct ion channel receptors are things like amino acid. It's like GABA and glycine, which tend to have inhibitory effects, which means they are going to bind to which type of channels. They're going to either let chloride come in or let potassium leave. There's also NMDA and glutamates. These are going to have EPSP effects or stimulatory effects, so they're going to open sodium channels. There's acetylcholine as a major type of neurotransmitter, and just to make it more confusing, there's two different types of receptors, and one of them is a direct ion channel type neurotransmitter, and the other one responds indirectly through G proteins. Nicotinic acetylcholine receptor is the receptor that receives the neurotransmitter from a motor neuron and stimulates muscle. Muscarinic, again, it works through G proteins, and this is a major receptor in the parasympathetic nervous system. You have neurotransmitter called biogenic amines, which are further divided into catecholamines like norepinephrine, epinephrine, and dopamine. And then you have indolamines like serotonin and histamine, which are related to things like mood. There's different peptides, so really small proteins. There's one that's called substance P that tends to increase pain, so if you injure something, you actually want more pain to that area sometimes. You want it to be hypersensitive so you don't use that region. Or it's also responsible for after you cut yourself and it swells and the area around the wound becomes more painful. That's something substance P is thought to do. Endorphins have the opposite effect, so you get like a runner's high, or endorphins are thought to be given off in cases of extreme pain, and they reduce pain. And then there's novel neurotransmitters like carbon monoxide, nitrous oxide, and ATP. So that's the neurotransmitters. So that's the umbrella metaphor, and basically I'm hoping that if you look at each individual chapter in this metaphor, they're fairly simple. The difficulty is putting them all together and making it make sense as to how neurons make decisions. And the first step of that is just how it makes sense in terms of the umbrella metaphor. And the next step is, can you apply it to basically other decisions that your brain makes? Whether it's a line that's vertical or horizontal, whether it's this word or that word. How do you apply these decisions to keep your car moving straight down the road? Or even more complex decisions about, do I go out to dinner tonight or do I go to class? On the test, I definitely would like you to come up with your own decision-making process. Don't just copy, do I take an umbrella today, and try and be a little bit more creative than, do I take a coat today? A common one is, do I go to class today? Again, you should be able to extend this metaphor up into higher order conscious decisions like, what are my political affiliations? What are my goals in life? How do I choose my friends? To simple cognitive processes like reading and turning left and following directions and things like that. Or, how do I feel right now? Do I feel like an idiot and I need to listen to this darn thing again? Or do I feel brilliant? Which is what I'm hoping for. Because you see that at the very base level of consciousness, neurons make decisions, and the processes are pretty simple, although they add up to pretty complex behavior. Or maybe you just go take a nap.